Hey, everybody. Welcome to episode 15 of the Reptile Rejects podcast. This week, I'm finally back after 3 consecutive shows of being away at Reptile Expos. So, let's get started. All right,
Yeah.
Dominic, how's your week been?
Well, first, thanks for showing up. It's nice. It's nice to actually have the host back on because, Shan did a great job, but I was starting to wonder if you were even going to come back. So, no,
No,
it's been
move
good.
straight to administrative duties.
Yeah, right away. It's been a good week. It's been decent. Let's go with what's new with me. Animal wise, I mean, not much. I'm trying to get a couple of last few big feeds in just because it's basically my last month of feeding. Everything's to me pretty good overall for that. Except for that, I don't know if I ever went into all the perches that I purchased that I made. Did I ever go into that in one of the shows?
I'm not
I don't know if
sure.
I did, but
Why don't you hit on that?
I did two things. So I made this guy, which is a Theris 3D, and it made me these little things here. I don't know if you can see what they are, but basically I built like 100 of these, which all will fit all these type of PVC pipes that are 1 inch that come with all the different elbows. So I can literally build all these jigsaw puzzles that'll be about like 4 inches high in the cage on the top so that I can still clean the paper, remove all the hides and all that without moving the perches. And if the animals are there, just more convenient. And I got these big monsters built. I don't know. Let's see if your camera actually can catch them. So
Oh,
these
yeah.
big monsters, water bowl, 2 levels, the top basically removes. Let me see, like that. Yeah, if it chooses to remove, well, it didn't really move. And the hide box in the back comes off too, and you can remove the top. Like these things are freaking amazing. They fit perfectly for all the little cages in the back, so I'm pretty happy with that. So I'm always all about convenience and time saving things and using the most amount of space that I can. And these things, they're always in the hide box or on top of them. So I literally just grab it, pull the whole thing out of the cage, move the paper out, put a new one, and I just literally stick it back in. And the animals just stay on there while I put it on the ground. So I hated those cages. They took me forever to clean because then I'd have to remove the branches and everything, and I just hated it. Um, except for that, man. No, life is good. Yeah, what about you? What's new with you? I think there's a lot new with you, actually.
Oh, yeah. Well, I need to bug Greg about building stuff like that because he should be able to build that up here with his little 3D printer army in his basement. But yeah, so with me, well, I went to Toronto, met you, met Shan, met Sean McCarty, I think is his name. I probably said that wrong. But I met a bunch of other cool guys up in Toronto, hanging out with my buddies from Alberta. We had a great time at Toronto. So saw lots of definitely saw lots of people there. It was a huge show with the dog part and the fish part, but then the big reptile area too, with lots of neat animals. So that was fun. And then ate some good food there as well. And then after that, they had our show here in BC. So I had to help put that on. So I had to take another couple days off work before and after and get everything all sorted and set up for Friday. And then help my buddy Tyrone vend that show as well, like I did in Toronto. And then we all hung out, had some good food again, stayed up late, talked a lot till 3 o'clock in the morning, and then another week of work. Sent my other buddy's animals back to his shop on Monday, and sent last year's holdback male Kentucky out to a friend in Ontario because it had a slight little tail deformity. So I was like, I can't hold this back for breeding, but I can gift it to somebody who's going to use it for educational stuff. So a nice bright yellow snake that has a bit of attitude would be great for an educational snake. So he has that animal now. And then this past prior weekend, I went to Edmonton, helped my buddy again because his kid was supposed to be at hockey tryouts. And then it turned out his kid was able to at least come on the Saturday. So there's the three of us all there, plus Greg and from Cornell's World was there. So we all kind of hung out that weekend too. Had a blast. Definitely more of a show like BC, maybe a little smaller, but they had a new venue there and it was almost like a plant greenhouse type thing, but like a nice, not like a full greenhouse, but like it was a greenhouse and like a nice area. It's hard to describe, but it was actually a pretty nice venue for what it was with many plants beside. If you liked plants, you would spend money there as well. So yeah, no, that was cool. Had a couple pickups. I finally pulled the trigger and traded and paid some money for some ring pythons from Cody Joe. So that was pretty cool. I picked up a 2024 female and a 2025 male from him. So that's cool. They've already eaten several times. They've already boxed up at me, although I haven't been bitten yet, so that's good. And then I also picked up a pair of sandboas, a nice albino male had anery, and a anery striped female had albino, just to add to the pair of Kenyans I already have that I've had for 14 years now. So I was like, I can breed these, so maybe I should add a couple more just because they're fun snakes, and it's always cool to once a year you get the 15 or 20 babies all there. So yeah, no, that's all that's new with me, I think. And so yeah, and then I had a litter of sandboas. I called it because I was like, as soon as I left for Edmonton, I like knew the female had her waxy poop, and I was like, she just pushed the substrate just a little bit, and like I wasn't fully committed, but then I was like, she's gonna lay, and of course she did. On Saturday, I texted my parents, and they double checked, and it was like, yeah, all 14 were already out, all on the bottom of the tub. So, and they all look good. There's no slugs left or anything. So if there was, she might have eaten them, but I kind of doubt it. So yeah, she
That's
had another
really
good
cool.
litter. And then I also had 4 subocs hatch out from my Wild Rose Pass pair. So that was awesome. The male was from Smitty that I imported a couple years ago. And the female I imported that from Rob Nimmo a couple years ago. And they produced 4 perfectly big eggs. And the babies hatched out perfect. And they are way bigger than I expected. And they're starting to shed now. 2 have shed, and the others are just starting to shed, and one's even eaten a frozen thawed pinky already, so they're already on the right track. So yeah, and then I've sold 3 Kentucky pines from that clutch of 8. I got 1 heading out tomorrow to Winnipeg, and then the other 2. Then there was 1 pair I traded to Cody Joe for the rings, and then I'm gonna hold back a male. And I think I technically have 2.2, but one of the females still has her umbilica kind of dried. And like I left it thinking it would fall off, and then it didn't. So I trimmed it down as tight as I could, but it's still kind of there. So it's kind of weird. So I'm kind of iffy. Like I'll see if it maybe sheds off or something, but worst case, she's just a pet. So I got for sure 2.1 Kentucky pines if anybody wants those. They're
Yeah, I
all
was gonna
feeding on
say,
frozen thawed.
I feel like a shed would take care of that, but it's kind
Yeah,
of
like
weird.
it didn't shed off in her first shed, but I don't know. She's eating live. Well, she took 1 frozen, and then the last couple weeks she hasn't eaten at all. So I need to, once the subocs are all shed out and the sandboa is shed out, I'll get a whole bunch of live from my friend and try to get them all going again and see how they do.
I get the feeling
So
you're going
yeah.
to be the guy that just picks up an extra 5 to 10 animals every show from now on. Your collection's just going to keep growing every time. You're
Hey,
going to
I
have
was
a nice
pretty
long
good.
list.
There's only 1
I don't
show I
know,
actually
man.
bought anything at.
That's
Out of
a
3, I
lot of
only
animals. I'm a little jealous of the
and
Bismarck, though. I'm not gonna lie. That one hurts.
yeah.
I'd want those.
Well, there's only 4 new ones. Well, and the Angolans from a month and a half ago now, but
Yeah.
I need to hold off, especially if this Bahama does go. Although I'm still iffy on that. Hopefully, she's not just like ovulating for the past 4 months, and I should have
Do I
thrown a
try to
male in.
guess
But
how many new snakes I've had now in 2025?
0.
Yep. Yep. Exactly. All right. Are you ready to bring on our new guest?
Yeah, already. This week, we'd like to welcome on Dr. Warren Booth from Virginia Tech and the BOA booth. How are you doing this week, Warren?
Oh, great. How are you guys?
Oh, very
Doing
good, very good.
awesome.
Thanks for coming on, Warren.
Yeah,
I've listened
thanks for
to
the invite.
so many podcasts with you on. I mean, you're like, for me, I was excited. I'm like, I love getting on genetic guys. Like, I know all these type of questions, and I'm like, I know he's a boa guy, but I know nothing about boas. So Michael's gonna go crazy on that, but I'm pretty excited to have you on. So yeah, thanks for coming on, man. I really appreciate it.
Oh, that's great. I hope I don't disappoint you. So, but I also keep a lot of pythons. So I don't keep ball pythons. but I keep a lot of other pythons as well, and a lot of calipers and stuff. So
Yeah, you had told us right before. I was so excited. I'm like, sweet, we can talk about pythons too. But I know Michael's got like a pretty. Actually, it's the first time he did a show outline, which I'm really impressed because I just usually wing it. So, but he's got like a really long amount of questions about many boas. So I think we're gonna have to go into the boa talk for all the other people that probably log on to talk about boas for you, right?
No,
So
that's great. I look forward to it.
Alrighty. Yeah, I think a lot of people have heard kind of your start in the hobby, but maybe if you could give us like a short kind of summary of how you kind of started keeping and how you made your way to the U.S. from Ireland.
Sure. Yeah. I grew up in a family where we always had a lot of pets. My parents owned a pet store for a while, and I remember when I was maybe 5, 6, 7 years old, sitting on our back step with a crate of tortoises. Probably they were probably Herman's tortoises, and we were picking ticks off them before they went into the pet store. I remember maybe at around the same age, in our bathroom at one point in time there was a 10-gallon aquarium, vivarium that had a redney tarantula in it that my dad was looking after for hens. So there was always animals around. But I was probably 15 or 16 years old, and I was helping my dad with his business. And we were in a house working on a fire and a boiler, and beside the fire was it must have been maybe a 20 or 30-gallon enclosure, and it had these fine branches in it, and I thought it was going to be fish, and because I was really into fish, and I went over to look at it, and there was no water in it, but there was like a garter snake or a ribbon snake kind of moving around these branches, and I just became kind of fixed on this thing for the 30 minutes we were there, or hour that we were there. And I went home and I asked my mom if I could get a reptile, and she said sure. And a week later I had leopard geckos, and then a month later I had a western hognose snake, and 3 months later I had a king snake, and it just kind of built up from there. And over a very short period of time, I got into emerald tree boas and Amazon tree boas, and some Sonoran boas and stuff like that there. And when I did my degree and my PhD in genetics and evolutionary genetics at Queen's University in Belfast in Northern Ireland. And in 2006, I moved to the US. Originally, the idea was to go for 3 years to work at NC State for a postdoctoral position, and 3 years turned into nearly 20 years. At that time, I sold most of my collection. I sold all of my arboreal stuff, but I exported 18, I think it was 18 or 20 boas and one ball python to the US to a friend in Delaware. And then I drove up and picked them up, and the collection has then gone from 18 or 20 to 130 or whatever I've got at the moment, of about 21, 22 species. So yeah, there's not been a year since I started in reptiles that I haven't had reptiles, and there's probably hasn't been a year in the last 30 years that I haven't been breeding reptiles. I think, yeah.
That's
Right,
pretty
nice.
cool.
Just by curiosity, I honestly thought you kept mainly only boas. So do you mind kind of going over your general collection just so we kind of know what we're talking about here?
Yeah, so with boas, I keep a lot of arboreals. So I've got Eastern Sanzinia, so the green Sanzinia, and then I've got a lot of emerald tree boas, Trinidad tree boas, which are Corallus schumbergeri, Amazon tree boas, Granada bank, which are Corallus granadensis. I've got annulated tree boas, hybrid emerald Amazons, and then I've got Boa Sigma, which are the Sonoran boas and kind of the Pacific coast boas in Mexico. I've got Boa Imperator, mainly Costa Rican and Nicaraguan, but I've also got West Snake K, Lagoon K, and Crawl K, kind of the island boas. I've got Boa longicauda, or Boa constrictor longicauda. What else have I got? Boas. I got a lot of rainbow boas. I got with Brazilians. I got T negative and T positive albinos, and then the regular. And then with Colombians, I got T negative, T positive, anarthristic, leucistic, and patternless. I think that's it with boas. With pythons, I got Timors, Womas, Darwin carpets, both albino and wild type. I've got children's pythons, T positive, patternless, and wild type. Spotted pythons, I've got Cape Yorks, and granites, and striped. And then I got the pygmy banded pythons, not the pygmy, but the pygmy bandeds. And what else have I got? I got Dunn's pythons. Uh, what else? Maybe something else there. And then I've got Howard's Draw grey banded king snakes. Got African high snakes. I got Western hognose snakes. I got a bunch of leopard geckos with my son. I got redfoot tortoises. And I'm probably missing some stuff there, but I think that's roughly around what I've got. So a whole bunch of different things.
Well, I was going to say that's quite the Noah's Ark. Just by curiosity, if you exclude any babies you've had this year, what's your number in your collection? Is that 100, 150, 200? I'm just kind of
It's probably about 100
ballpark.
About 130, 140, somewhere in there.
Yeah, that's a pretty good. That's pretty
way
big.
Too many. Yeah. I mentioned Timor's, right? Yeah, and I've got desert king snakes from Portal Road in Arizona. Yeah. So yeah, too many.
Nice.
But I keep saying I'm not going to get any more, and then all of a sudden something else appears in a trade or whatever. Or else I hold back stupid numbers of what I produce.
That's awesome. So like because in Tulsa you were able to keep like most of your arboreal stuff at your lab. Now you've had it moved all into one spot, I'm assuming.
Yeah.
So
yeah.
How does your room kind of change? Like how did you have it set up in Tulsa versus how do you have it now, and what's
so
kind of changed with that?
In Tulsa, I lived in a house that was just over 100 years old. My basement was partially finished, but it had all of my racks. At that point, I had Freedom Breeder racks, and then I had a bunch of arboreal enclosures. And I kept most of my arboreals there, except for a lot of my emeralds. I kept a lot of my emeralds at the lab, in my office at the lab. When I moved here to Virginia, to Blacksburg, the building that my lab's in is this ultra high tech, ultra biosecure building. So having animals in it is not the greatest because of the potential for DNA, airborne eDNA, and I still do have 3 emeralds there. I've got 3 baby emeralds in my lab, in my office, but I've got everything here. So the nice thing, we build a house, so we've got nice climate controlled rooms. And I basically got 3 rooms that I've got the animals split between. The arboreals are all in one part, the terrestrial stuff's largely in another, and then I got another room for babies.
Nice.
Yeah.
So I like for work I like drywall and steel frame new residential houses. How have you? Do you find it's almost harder to? Obviously, like the temperatures are more consistent in the newer home.
Yeah.
But also, I find the air tightness and all that is a bit more strict as well. So air movement the over, like you can't drop it as cold kind of thing. How is that affected, or have you seen anything yet?
Yeah, so that's a big thing because in my former home, everything's in the basement still, but the basement I could get down in the winter. It got down to about 60 to 65 degrees. Here, it's hard to get it down below 72. Which is fine for everything I breed except for the Eastern Sanzinia. So this year, I'm going to be moving them to. So this the basement it's a walkout basement, but it means that 3 sides of it are pretty much covered, with soil, up the sides, so they're really well insulated. So I'm actually have moved a large enclosure right beside the window at the coolest end, where I can actually crack a window open a little bit. And I'm going to see if I can get the temperatures down into the 50s. We'll see. The temperatures certainly will get into the 50s outside and lower. I just don't necessarily want my entire basement sitting at 50 degrees or 55 just so that I can get my Sanzinia cold. So it's going to be a new one for me for that. We'll see how it works out. But it's moving from a home that was very easy to get cold to a home that's not easy to get cold at all is a totally new thing. It also works, with my boa sigma, the Sonoran boas. They like to get into the low 70s or high 80s, high 60s, and it's hard to get my room into that temperature as well. So yeah, there's a bit of playing around with doors being open and fans blowing and stuff like that there. So and also modifying how in Tulsa, I could leave my hotspots on permanently. I never modify the temperature. but because the room dropped so much, it meant that the front of the enclosures always got down to the low 70s or the high 60s, and the back of the enclosure still had a hotspot. Here we can't do that, so it means that I'm actually putting, the heaters on at least some level of timing, where there's night drops, and I have to work out when the night drops start to be able to get down to the lowest temperature and still be able to warm up again. So there's a bit of a learning curve for that there, but last year it worked out really well for breeding. We produced a lot of babies. This year there's things that I want to produce that need to get colder, so that's going to be a new kind of process. So we'll see.
That's interesting. Yeah, actually the last couple of weeks, because Cody Joe's been at all the same shows as us, chatting with him. He actually has a idea. So apparently, the old zookeepers at the Calgary Zoo back in the 2000s, they used to breed Sanzinia every single year, and they never dropped those animals below almost 80. They just stayed ambient at 84 the entire year, and they dropped babies every year apparently.
Yeah,
which is definitely different from what we've all heard, but right. Apparently that's how they bred them, which is really weird.
I've got a friend who used to be a zookeeper at the Bristol Zoo in the UK, and every year they would produce green Sanzinia, and I don't believe that their reptile building ever got below at least 70, but that part was like an old Victorian building, and drafty and stuff like that there, but I still can't imagine them getting into the 50s easily. So
no,
it's going to be a learning curve, but we'll wait and see. I think green Sanzinia are still one of those animals that people haven't worked out fully, and I'm not convinced that they need to get into the 50s. And I need to really look at Madagascar and the temperatures in Madagascar and see how they regulate throughout the year. But I can't imagine them getting into the 50s too often for prolonged, like for 3 months at a time. But maybe I'm wrong. I need to have a look.
Maybe it is just a night drop, and then they get back up to 80 or something.
And light cycling and stuff like that. There, food cycling. Maybe they need a few more triggers. A lot of animals just require one. You can cycle temperature, you can cycle light, you can cycle humidity or food, and very often if you do one of those, it works. But maybe they need a few more of those triggers.
I often wonder whenever I hear people doing like those species that really have a way of doing it, that everybody does have success by, let's say, bringing it down to 50. Some people are like, well, I never bring it down to 50 and it works perfectly. My thought is probably those people have had those animals forever. Yeah. And they've gotten them so used to their room in such a certain way that after 6, 7, 8 years of growing them up in an environment where it doesn't drop to 50 and all that. And I'm thinking like the zoo or things like that. Sometimes I wonder if it's just they got their animals to cycle and get used to that environment, and those animals never ever see that. And like you're saying, all the other triggers is what makes them go, right? It's most likely food cycling. It's most likely light food and other things like that. That's my reasoning. The only problem is, I feel like it's easier to do it with all the triggers that what. So to me, I wouldn't remove a trigger like temperature cycling, because to me that's the easiest one to do, and it's the one that always works.
Yeah, I'll definitely be able to get them into the high 60s, easily. I can do that there. And I can definitely have them with a light cycle. And I can definitely have them with food cycling. I'm starting to cycle them now food-wise. But there's no way I can get them into the 50s, because that means that room in the basement, which is a large area in the basement, is going to get done. And I don't think my kids are going to be too happy about that. Their playroom, they're just, sitting in sub-zero temperatures. So we'll wait and see. I've had them since they were babies. They're 5 or 6 years old. And we'll see how they go. If they don't go, they don't go. And next year we try something different.
How many cages do you have that you want to drop in the 50s?
Just one. So I put a 4 foot by 3 by 2 right in a room in my basement, which is right beside the window, and I've changed it all out so that the hot spot is actually only on one corner on a shelf, so they can go up there and get heat, and the rest of the time they can go anywhere else in the enclosure. So, and I put it right on the ground, so it's that part of the basement we haven't finished yet. So that it'll be on a concrete pad, so they can't get onto the concrete, get close to the concrete kind of thing. It's going to be right on the ground. So we'll see what temperature they get down to. I'll put a large water bowl and just see what they do.
If you can't get it cold enough, like I have the same type of issue where I'm in a basement, but my room goes down to 64 is the lowest. I can't get it lower than that. And I
Yeah.
figured that the best solution is simply to drill a 4-inch hole, like just for duct pipe, attach it to a cage, and put a fan in there on a
Right.
timer,
Yeah.
and
Yeah.
or on a thermostat. It's to me, it's a solution that works if you only want to cool certain cages.
Yeah.
But
Yeah. Yeah. No, I think that's a great idea. And as long as they've got that basking spot where they can still get up to and return to, in the 80s to bask to counteract any, ultra cold temperatures, I think they'll be fine. So I'll just give it, I've got a bunch of little Govee monitors, so I'll just monitor it and see what it gets down to. And,
Yeah.
I can't imagine that they need to get into the 50s, but, that's worked for people. I think the big problem with Tanzania, you guys spoke to Bill Hughes a few weeks ago, and you're talking to Bill and talking to Keith, who have bred them. They get them cold, but I've also talked to other people that haven't got them cold, and they produce healthy babies. So I think for me, it's a species I haven't bred before, so it'll just be a total learning curve. I just don't want to lose any.
With your luck, you'll probably get them to go first try.
Green Tanzania aren't easy to come by. So as long as they are alive, then if they don't go this year, then I can try again next year.
Yeah. That's why it'll be interesting to see when we get Jeff Murray on, because he said he would come on when he has some time. So I definitely want to hit him up, because he's a big Tanzania guy, if I recall. That's how he pays
Jeff's
for
got
everything.
He's got a lot of stuff. He's got a lot of Tanzania, a lot of Corrales,
Oh yeah.
a lot of the Caribbean boas. Yeah, Jeff's a great guy.
I assume that's where you got your Grenadensis from.
I did. Yes, I got my Grenadensis from Jeff. Yeah, and then we did the import for the Trinidad Richenberger eye.
Yeah, that's awesome. The Grenadensis are like definitely something probably nobody in the normal herp hobby knows about, because obviously, and then you got you got those guys, and then you also have Cook Eye, which I believe Jeff has too.
Yeah, he's got
like true Cook Eye.
Yeah. And nobody's got those. You see people list them on price lists. Corrales Cook Eye, or Cook's Free Boas, but they're not. The Cook Eye come from a Lesser Antilles. They're really hard to get, and they haven't been brought in for many years. And also they're while they superficially look like Amazon tree boas, and to most people they look like Amazons, they're very different. Especially the babies, just like the Grenadensis, getting them feeding, they're lizards focused. Grenadensis seem to grow very slowly. The ones that I've got here are really slow growing. They eat really well. They just don't put on size or weight, comparable to what a Hortolana would do, or a annulated or whatever, But they're really cool. The Grenadensis, there's a lot more pinks in them, a lot more greens. They're a cool snake.
That sounds similar to the.
They're awesome to have.
Sorry,
it's
you
like
keep going.
Trinidad's, but it's the big pain is whenever they come to feeding babies. Lizards are a pain in the ass, or lizard sending, or all that whole process. It's not like just a nice good old boa constrictor which you can get going on a defrost, mouse, and it's no hassle. Whenever you got these things, you get gray hairs pretty quickly from the stress of sitting playing with them.
Sounds a lot like the Chilobothris stuff, where like you get them going and then they just take forever to grow. Like I got a 4-year-old male Bahama boa, and he's 4.5 feet.
Yeah. They just don't seem to put on a lot of size.
And like that thin.
Yeah.
Like he's growing a lot since I got him, but it's that slow and steady.
Yeah,
I can see why
because I see them every day, right? So you don't really notice them growing as much, but they really just take forever, But they're beautiful snakes, So.
Yeah, I can see how the lesser Antilles or any of the Caribbean kind of species take. They probably shouldn't even breed till they're like 8. Like they like that. Like even 6 seems kind of young for them.
Yeah, I think lizards, and then they switch to birds, and it just means that it just takes time, for them to really put on weight, and they don't need to be big.
No.
I think about a lot of the Amazons I used to have whenever I bred them 30 years ago. They were 6 feet and eating medium to large rats. They're huge. They were way beyond what an Amazon should be, and dropping like 25, 28 babies. And then I see people breeding Amazons now, and they're tiny. They produce 5 or 6 babies, and I think that's probably closer to what they should be. Um, but the Granadensis, I think they're as adults probably they are smaller in the wild. But yeah, they're fun. They're cool snakes. They're feisty. Just like the Trinidads. They're not like Hortolana, which are just floppy little animals that try to fake strike at you and fall over. Like Trinidad's, just like the Venezuelan Richenberger, right? If they see you and they want to bite, they know where to go. They're like scrub pythons, and Granadensis is somewhere in between that. They're not they're not happy little snakes. Whenever you open up the enclosures, they heads up and they want to kind of bite at you. But they're really cool. And I suppose because I've had them for a while, the novelty's gone from them. And yet I think back to 25 years ago, I would have cut off my hand to get Granadensis or Richenberger or whatever like that there. So I think I think that's the hobby. But once we get it, then our sights get set on the next thing. How can I get X or Y or Z?
Although there's those species that you get, and then you're like, I'm glad I got this, and I don't think I can live without this species anymore.
I feel that there with any of the Corallus that I've got. Like I'm a big Corallus nut, and if I had to keep anything, or I could only keep one kind of group, it would be the Corallus tree boas. I really like those a lot. But because I look at my Eastern Sanzinia, and what are they like adults now? You can't even find, but if they were, they're probably 5,000 or 6,000 each. And you pick them up and they shrink up into a ball, and you're like, all right, it looks like a ball python. You set it back down again, and just wait. It's kind of interesting to see that these animals that people really want, and whenever you actually have them, you start looking at them and seeing the little issues that they have, where they're like, oh, it's not as exciting as I thought it would be. And then you turn around an hour later and it's sitting in a tree and it's wanting to feed, and they're just really cool.
Oh yeah.
I find like there's a lot of those species where you never thought you would get, right? Like I don't know, if I look like the top cage on the right there, that's my rough scales. Like I remember starting, I never thought I'd have that. Or even when I started keeping carpet pythons in the 90s, never thought an albino would ever be available.
No,
and you look now, it's like not a throwaway morph, but it's like the most basic morph in carpet pythons.
I totally agree with you. Like I do think about that. Because I remember years ago in probably the mid 90s, seeing in Dave and Tracy Barker's book where they had the picture of the albino Darwin, and I was wow, that's amazing. And now Dave and Tracy are friends of mine, and I have albino Darwins, and it's just like, and I sell them for like $400 a piece, and it's like,
it's crazy.
It's I sell them for a fraction of the price of what I sell emerald tree boas for, which is just amazing considering how rare they were. It's and just like Trinidad tree boas, that's for me. That's always been the pinnacle of Corallus, because they're just huge, big scrub python-esque and impossible to get. Now I've got a whole army of them in my basement, and that's the one I go down and look at every day, and I'm still like, whoa, I can't believe I actually have those, because if I lost them, it'd be virtually impossible to get. Thankfully, we split the group that we imported among 3 of us, so if something did happen, there's that kind of backup. But if they all went, then there's no more in captivity. Right, you can't find them. So it is amazing to have those animals that you didn't expect to have. It's like Angolan pythons. I had Angolan pythons for a very short period of time, and I regret selling them. But whenever
I don't.
But in the 90s, Angolans were, animals coming out of like the Transvaal Snake Park that were, $40,000, $35,000, a pair. It was just insane.
The Angolans a joke between us, because I got him, I bred him a couple of years, and then I just grew so tired. Every time I look at him, I just saw a ball python. I just couldn't do it anymore, so I had to let him go. And as I was letting go, Michael got some. He's so excited about it. I'm like, yeah, you could have him.
Yeah, I think they're really cool. They didn't have the temperament of a ball python.
Sure.
No.
For me, they were
no, they don't.
Of an angry blood python. Mine would bounce off the cage floor. Like it's the only ones. Those and some blood pythons that I had would actually propel themselves off the cage floor and strike out at the same time. They were not the happiest little beasts that I have, but I still loved them. But I was buying a new truck at the time, and if I sold them, then the extra money I had for a down payment would bring down my monthly payment. I thought, yeah, I can pick up Angolans again, and I haven't. That's 6 years ago, but I maybe at some point I will.
Yeah, I'm sure somebody can hook you up with a
I'm
nice
sure I
pair
can
of them.
get
There's
some
some
trade.
really nice Angolans out there now.
Yeah, there are some great ones. Yeah.
Yeah, I don't think I'll ever get back at Angolans, but they are pretty. Mine were tame. We had I had a trio, and they were puppy tame. I had no issues at all with them, so no issues. All right, so
Yeah.
I'm going to go into more of my geeky questions. I got one that I really would love for you to explain. So, more about the sex chromosomes of snakes. I'd love for you to explain, like, evolutionary. Why is it that some are XX and XY, some are ZW, and I don't know what the other one is, ZZW.
ZW. Yeah.
There you go. So why is it? And is there another type, or is that the only two types?
That's it. From what we
That's
know
it.
of snakes. Yeah.
So what is which family or groups are which, and what's the advantage? Like, I'm trying to figure that out. Like, why are some so different than the other ones, and how did we get there?
Yeah. What's the advantage is a good question, because I really couldn't address that there. What we thought for a long period of time was that snakes, like birds that we know of, were ZZ and ZW sex chromosomes. And what that means is that the males are ZZ, the females are ZW. This is like the opposite of mammals, right? So mammals are XXXY. The female is XX, which means that she can only ever produce an X chromosome. So she can only ever produce essentially a female X chromosome. The male determines what sex the offspring is going to be because they're XY. They can either give an X or a Y, right? An XX being a female, an XY being a male. In snakes, we thought that they were ZZZW, where the female is ZW, the male is ZZ. So therefore, the male doesn't determine the sex; the female does. Well, pretty much the advanced snakes, from the Acrochordus, the file snakes, the kind of like the elephant trunk snakes, from there onwards. So all the colubras, rattlesnakes, pit vipers, elapids, they're all ZZZW that we know of. We got into this because of parthenogenesis in snakes. In my lab, we did some work on we've done a lot of work actually on parthenogenesis in snakes, kind of fell into it. And we got these contrasting patterns between colubrids that were producing parthenogenetically and rattlesnakes and elapids and the boas and pythons. And what was happening was, with parthenogenesis, the boas and the pythons were producing only female offspring. Whereas in the rattlesnakes, the pit vipers, all that stuff, they were producing only male parthenogenetic offspring. And they were producing a whole bunch of infertile ova. In the boas and pythons, you didn't get them. And we started trying to work out in our minds, myself and my collaborator, Gordon Shewitt. Many people know the Shewitt line for rainbow boas, for carpet pythons, for Hog Island boas. Gordy was a professor at Arizona and in Georgia. He now works with Bob Ashley at Eco Universe, and there's guys out in New Mexico. He had done work on parthenogenesis in snakes for a couple of papers. I got involved in doing another project and we kind of got together. But we were finding that, why are boas and pythons only producing female parthenogens and everything else is producing males? And the most parsimonious answer was that the sex chromosomes had to be something to do with it. We did some work with a friend of ours, Tony Gamble, and we actually sequenced genomes and we sequenced sex chromosomes, and we found out that boas and pythons were actually XX, XY sex chromosomes, just like mammals. And oddly, they evolved that mechanism separately. People don't realize that with boas and pythons, they're very old snakes, a very old lineage. Boas are about 110 million years old. Pythons are about 50 or 60 million years old. So pythons are like half the age of boas. But they evolved these sex chromosome differences independently from everything else. But they're XXXY. The only ones that are weird are the Madagascan boas that we know of so far. The Madagascan boas are ZZZW, which is why that is. And don't ask me, it's just weird. Candoia quite probably could be ZZZW as well. But what's the benefit of it? I really couldn't tell you. It's just a term. The ZZZW XXXY is just It's just it's just a term to be able to just tell you whether it's the females determining the sex of the offspring or the male. But sex chromosomes across snakes are weird. Some of them we find the XX and the XY, or the ZZ and the ZW, don't change in size, but both the exact same size. We find some of them are totally different. Just like with humans, the X chromosome and the Y chromosomes are totally different sizes. The Y chromosome is really tiny, the X is really big. We find in many of the colubrids the same story. Right? The Z is really big, or the W is really big and the Z is really small. Or it's vice versa. Z is big, W is small. Sex chromosomes are just really unusual, and don't even talk about lizards at that point, because you can go through lizards and you can find some some families of lizards where they all have in the same groupings they could have XX, XY, ZZ, ZW, or temperature sex determination, and therefore no sex chromosomes. So yeah, sex chromosomes are weird, and we touch upon our work with them a little bit here and there. We've had papers to identify them, but we don't really go into the evolution of sex chromosomes. My friend Tony Gamble, who does some really great work on gecko genetics, and you should get him on. He's a great guy. I can give you his details. He really is a sex chromosome kind of guy when it comes to research.
Very cool. Do you mind if I just go back to one thing you said? You said that pythons and boas evolved the XXXY separately. Does that mean that they were ZW before and both along the way came down? Or what are we thinking?
Probably a common ancestor between boas and before that, and so on, quite possibly could have been. What we don't know is that there hasn't been enough sequencing of sex chromosomes of the snake phylogeny to know in those basal lineages, so from boas backwards towards the into typhlops and stuff like that. There, we don't really know what sex chromosomes that they're utilizing. There's been work that was done based on cytogenetics and just basically smashing chromosomes and looking at them and sorting them, but we still don't really know what sex chromosomes a lot of these snake families use, which is fascinating. Tony Gamble, I think, is working on a lot of that. As part of the work, we just recently started sequencing the boa constrictor genome to chromosome scale, and the carpet python genome we're doing to chromosome scale with Tony to start understanding sex chromosomes, because Morelia are really, really weird when it comes to sex chromosomes, which is why producing genetic sex determination is really difficult for Morelia, which is why producing sex determination for green tree pythons has been a bit of a nightmare. So therefore, to get around that, we're actually sequencing those species to chromosome scale to try and understand that. Not to try and determine, to be able to make a sex determination test, but to be able to understand the sex chromosome evolution for them.
So I've heard that many times about how sex determination in Morelia, especially chondros, just doesn't work or is really difficult. Do you mind explaining why they're so different, or they're really weird? Like, I've heard that term many times, but what's actually different? Like, are they still XXY?
Yes.
Okay.
But there's just weird things going on. I can't really talk about it more because it's part of a larger project, Tony.
Okay. No worries.
But it's not to the case where we could simply identify a marker easily on the chromosome and say, right, that is a male marker or a female marker. There's a lot of weird duplications, a lot of weird unusual things happening with the sex chromosomes. And probably within the next year, there should be stuff out, year and a half, there should be stuff out about that there. But yeah, they're not straightforward.
And one more thing related to sex chromosomes. So when it comes to morphs, a lot of the morphs are in complete dominance, are recessive and such. Now, when it comes to humans, a lot of illnesses are related to sex chromosomes and are tied to sex chromosomes and can be passed down that way. And therefore, the XXXY is going to have an impact on diseases passed down. I've always wondered, do we have any actual mutations or morphs, or even, I guess we could go down to disease route too, that are related to our sex chromosomes? Like, is that possible that you'd have a mutation that is tied to either only the Y, for example, so that only the males could carry it and such?
Yeah, we do. In ball pythons, the banana and coral glue is a sex chromosome related mutation, which is why you breed a male banana to a female, and you produce only male bananas, and then one in every, 25 or 30 will be a female, and that's because that has jumped to the W chromosome from the Z, so you got recombination occurring. But that is the banana in the coral group is a sex determined is a mutation on sex chromosomes, which is why you just get the sex bias. So if you have a mutation where you breed it and you're primarily getting one sex, it suggests that it's on a sex chromosome. I'm not sure with other species, but the banana ball, the coral glue, which is the same thing, they were the ones that kind of came up early on, and which is why the story was so convoluted with those early on. People were like, well, they're producing these animals, but they're not releasing females, or they're not releasing males, or whatever it was, and it meant that a lot of distrust was built up among breeders. Are they holding back all of these ones and not releasing this sex, and so on? And it turns out it's a sex chromosome related mutation.
That's kind of cool. Actually, I think I had heard it about ball pythons, but I'll be honest, I know nothing on ball pythons. So it's the one thing I really don't know anything. But it's interesting that you could have morphs that are related to that.
Yeah.
Very cool.
Yeah, you have to realize that a lot of the pathways that we see to get to a phenotype, whether it's striped or whatever, it's a long chain of events. It's not just like turn this off and all of a sudden you get that. There's a long chain of events where things could happen anywhere along that pathway, and that eventually gets switched off and you get the phenotype. So these things can be on different chromosomes and so on, these different pathways. So if one changes or one doesn't, you can get a totally different phenotype. So we still don't understand mutations in snakes very well. With the work that's been done by RGI and the rest of the ones that are doing the shed testing and so on, now that we have a ball python chromosome level genome, we can map mutations to it and start understanding where they are. We're kind of doing that with boa constrictors, and in time we'll be able to determine how many are sex chromosome related, how many are not, and so on. Yeah, I'm not aware of any in boas that are sex chromosome related, but definitely in ball pythons there's one, and I think there's something in colubrids that I've heard it's primarily a bias in the sex.
Oh, is it the Mex? Isn't the granite gene
something like
somewhere?
Yeah.
Yeah, I remember John
there's a
Lasseter
colubrid
talking
window.
about that.
Yeah,
Yeah,
it's kind of cool.
I forget if it was like if the granite or the super granite has the red head or something, it's always a female or something like that.
Yeah, and the thing with that is that in order to understand that, we then need to sequence those species to chromosome scale, which is it's a lot of work, and then identify the sex chromosomes, and then identify the mutation on the sex chromosomes. Yeah. And there's no money in that, right? It's not a research project. We can't go to the National Science Foundation and say, hey guys, I'd like to get $500,000 to study, mutations in ball pythons. It's not going to work, So this is all the work that is all hobby related. Like it's literally, it's my own money going into it to fund the research, so that's all be done on parthenogenesis and stuff and sperm storage. It's not being funded by any research council.
So I'm curious because I remember you talking about in a past podcast on how, so obviously the Madagascar boas are like their own thing, basically, kind of like how sand boas and rosy boas and rubber boas are not fully boas either.
Yeah, they're actually more basal.
Yeah. So are pythons and boas the only ones that actually have like parthos will be female? Because I remember you talking about how Madagascar boas, the parthos should be male based on their chromosomes. Same with lapids and all that stuff.
All of the parthenogen Madagascar boas that we have in my freezer are all males. So far for what we know. But again, there's a couple of reasons why we don't know this. One, a lot of the species that are more basal to the boas and the pythons, we don't know. So the ones that are more old in terms of more distantly related, they're not kept really in zoos or private collections. So therefore, the likelihood that we see parthenogens is slimmer. And also, to be able to find a parthenogen, the reason that we find so many in boas and pythons is because of mutations in captive care. People are keeping large groups and they're breeding A to B and they're expecting C. and then but they're getting D, right? And then they realize that combination can't produce the offspring that they see, and then so therefore there's visually something that clues them in immediately. If you have a zoo and they've got a pair of Xenopeltis or some of the older, more basal boas or pythons, and they produce eggs or offspring, well, there's a pair there, so therefore they have to be from those, right? They could be parthenogens, but because they're not visually different, there's no reason why they would expect them to be parthenogens. So we've been doing a lot of testing for zoos to test for sperm storage or parthenogenesis, but we just don't have those data sets built up for those other species. So until, if we started finding parthenogens and we could determine the sex of them, that gives us an indication of what sex chromosomes they actually utilize. But for those older species, we just don't know anything. Even with boas, right? We have parthenogens in boa constrictor, in parata, and boa sigma. We have them in the Corallus group. We've got them in rainbows. We've got them in the Chilobothrus. We don't have any in rosies or sand boas. At least we don't have any samples, and I haven't heard of any records. We've got them in the Sanzinia and the Madagascan boas, and then with pythons, we've got them pretty much through a lot of those. But again, it's all the ones that are being kept in captivity.
That's really cool. So I got a bit of a weird question here. So when it comes to pythons and boas, the females are XX. So I totally understand why they're a half clone. They only produce females. That only makes sense. They can only pass down the X. They copy the X twice. They're an XX, right? But when it would go to, let's say, colubrids or all the other species of lapids, that it would be the females would then be ZW. Now,
right.
If a ZW doubles up, you're either doubling up the Z or you're doubling up the W. Now, my guess is, why is the WW not producing an offspring? It's a weird question, but I'm just trying to figure it out because I realize it's obviously not a sex, but technically you should have a third sex then in that case, right?
So that's a great question. And actually, in some fish parthenogens, you find WW. They call them super females. And they can be induced through pressure, temperature modifications, and so on. With fish, with sturgeon, and stuff like that. With snakes, we don't see it. The W chromosome is that highly reduced. It's not having its whatever complement chromosomes that are needed are not there to have it in the WW form. The ZZ, however, is totally viable, which is why we see with those advanced snakes, the pit vipers and the elapids and so on, they'll produce a handful of parthenogens and then a bunch of infertile ova. The infertile ova would have been the WW, and the offspring are the ZZ. And what we also so what's happening is also they're not doubling that X or that Z. Then whenever sex cells are produced, cells dividing, works will produce every sex cell will produce 4 sperm. But when the sex cell divides to produce eggs, it produces 1 egg and what's known as 3 polar bodies. So basically, they're cells that will degrade and will not be used. So you get a sex cell divides in 2, and then they divide in 2 again, so you get 4, right? These ones on this side, they're not going to do anything. It's actually the ones on this side that will actually result in the parthenogen. And those 2 contain a set of chromosomes that are almost identical. Not fully identical, there's slight variation at the chromosome tips, but they're not fully identical, but they actually fuse together. And they're either fusing secondarily or else they're not separating. One of the 2, we don't know which one yet. But that's resulting in the egg now having its full complement of chromosomes, and it's believing it's fertilized. I believe, and we from other work that's been done on fruit fly genetics, we believe that they just don't separate. So you go through that first round of division, and then where you should divide again, they just don't divide, and therefore that cell contains the 2 Z or 2 X chromosomes, and will then go on to produce parthenogens. We see it in crocodiles, we see it in birds, we see it in sharks. It's actually quite common across many systems. We just don't see it in mammals.
Any super females in any other reptiles? Does that exist in any or not in the reptile world?
Not that we're aware of. Yeah, not that we're aware of. But again, the whole parthenogenesis thing, there's no money in that research, right? So it's all It's all being done just by labs that are doing other stuff, and they're kind of dip their toes in it. You know, we originally believed with the first paper we published on parthenogenesis in boas, which was in 2011. So what's that, like 14 years ago? We thought that the females were actually WW because we believed at that point that the boas and pythons, just like all other snakes, were ZZZW. It was only later on down the line that we worked out that they weren't. But we're not aware of any that are those WW super females. We don't know. In time, more genome sequencing will address that, and in time, with more zoos and so on coming in online with testing samples, then we might find that to be the case. But so far, we haven't seen it. And also, across the snakes, even those with the ZZZW, we don't know the level of differentiation of the Z and the W chromosome. Right? How different are they in size across all of those snakes? There might be a group, just like the weird Madagascans that have got ZZZW instead of XXXY. We might find some of those colubrids or some of those pit vipers or elapids that have undifferentiated sex chromosomes, and you theoretically could have a WW or a ZZ. Yeah, we just don't know that yet. Yeah, it's still, the whole parthenogenesis story is still something that's still pretty new and pretty novel, even though the work that we've done over the last 15 years has revolutionized it in many ways. We still don't know a lot. We're still at that tip of the iceberg, and because it's purely hobby research, it's not what my lab does. It's stuff that we do on the side. But we advance it in little bits here and there. We sequence a new genome and we can learn where recombinations occurring, or we sequence another genome and we can learn a little bit more. And we keep, people keep sending samples, so we build up this whole database. And then, whenever I can add 25 samples to a sequencing run of other stuff that we're doing, it just absorbs into a different project, and we get that data and set it to the side and then play with that later on down the line.
Yeah, that's fascinating. I always liked when you go into the chromosome talk and stuff, because that's just super cool to hear.
It's
I should
have a whiteboard. My wife would kill me
if
I wrote on this wall.
I love it.
But a whiteboard would work better, If we're in my
I actually thought it was a whiteboard behind you.
No, and it's going to be, in about 2 weeks' time, it's going to be a bookshelf that I'm building along here.
You got to put your book collection up.
And that means maybe I could actually write on it, because it's going to be a bookshelf soon. I don't think my wife would be happy.
Yeah, I think if it was
paint
only
over it, it'll be fine.
Yeah, I think if it was only me hosting, I'd be going into these geeky science questions because I find it so interesting. But how about we switch to Michael? You want to go down one of the boas you want to talk about?
Sure. I just have I think I have one more question.
I'll go for it, man.
Those Bimini blind boas.
Yeah.
What are they like? Because obviously, I'm pretty sure they're asexual. So are they
they're
in a different
asexual
category or something?
So they're triploid. So they have, whereas we have, for example, XX, they would have XXX or XXY, or they're a triploid system. They're derived from hybridization. So you see that in lizards a lot, right? So a lot of the whiptails and some gecko species and so on were two species of bred, and they produced offspring. Instead of having diploids, so XXXY or whatever, they're XXX or XXY. And they therefore can't produce sexually, they can only produce asexually. The Indotyphlops, the brownie blind snakes, they're completely asexual. I read a paper years and years ago where they said they'd found a male, but I wasn't able to find any confirmation of that there.
It could have just been a different species of blind snake.
It could have been, and it could have been a bunch of different things, but more than likely it was a different species of blind snake. Yeah, because those things they're tiny.
Unless you have a microscope.
I used to have a whole bunch of them in my lab in vials, and they're just like thread. They're like a piece of yarn. It's just tiny little things. So being able to sex those is you have to be pretty good. Yeah, but they're unusual.
Yeah.
But that means that's why they're so successful as being one of the most widely distributed species on the planet. Right? They're also known as the flower pot snake because they were found or distributed by nurseries that would be selling plants, and they would actually be in the base in the soil, and they go from one place and they're shipped halfway across the country or to a different country, and then they come out of the soil, and then they produce parthenogenetically and establish a new population elsewhere.
Do we know how they became like their own thing? I guess because they're from the Caribbean,
I believe so.
or
Yeah.
okay,
And yeah, I don't think there's been enough work really on the whole Indotyphlops group, that genus, to work out which ones came together to form that hybrid. I'll need to look. I haven't actually looked recently. There might be some work that's been done recently, but at least in the last 5-6 years, I can't recall seeing any papers that came out about that. There again, it's one of those, you have to have the money and the will to do it, because it's not something that's going to be funded by a research council
yeah,
unless it's built into this bigger story of, like, we could probably do stuff. We do a lot of work with invasive species, and because it's a highly invasive species, we could probably develop work relating to it in that sense to address that question. But again, it's all based on the science and where the
yeah,
funding's coming from. It's not easy to get funding for science. With the work that we do, there's about a 4% chance that your funding, your research project will get funded. And whenever it takes you 3 to 6 months to write those proposals, it's a pretty difficult thing. So therefore, the likelihood they're going to turn around and go, yeah, I'd really like to know where Indotyphlops came from, it's pretty slim. So we build it into our other little projects here.
So if
I just found it neat how, like, looking at West Texas stuff, and you're like, well, you can find 3 different blind snakes out there, but you need a magnifying glass to determine which species you actually found.
Yeah. And DNA is even better for that. Yeah, that's right.
So if we look back at all the snakes, almost every single snake, actually pretty much every single snake except for the blind snakes, are diploids, right? So the fair assumption would be to say that every single snake derived from snakes that were diploids, right? The ancestors would all be diploids. Absolutely. So then if the blind snakes are triploids, that would tell me that they're the ones that had probably like a mutation or some kind of a problem in the replication of their DNA, and somehow one of them survived being a triploid or no?
For them, no. It's actually a case where most of the blind snakes are diploid also, and that then 2 species that are unrelated came together and bred, and because of chromosomal differences and mismatches, that they produce triploid offspring. So there's been some really cool work by Bill Neves and Peter Bowman on whiptail lizards, where they could actually cross these different species together and produce hybrid offspring that were triploid. We find a whole bunch of those triploid hybrid species in across the US.
Is that the 6-line race runner?
Yeah, a whole bunch of those. Yeah, those race runners and stuff like that. There, yeah.
Oh, that's really.
They tend to be evolutionarily very short-lived species. In the tens of thousands of years, not in the millions of years. Because they're not producing sexually, they're not getting recombination occurring between genomes, and therefore they accrue mutations, but they don't remove the mutations, and therefore over time those lineages build up more and more mutations that are detrimental. And then they get, like, eventually there's what's called Mueller's ratchet, where it just kind of knocks them out. Whereas if you have sexual reproduction, you've got this outcrossing occurring, this recombination and mixing up of genes or of chromosomes, kind of, and you eliminate many of those mutations.
Interesting. Yeah, that's pretty neat. Oh, I have one more question, because I just remembered, because I keep subox, and obviously they're in a genus with another species.
Yeah.
Oh, nice. Yeah, they're fun. I love mine. I just hatched out the 4 wild rose pass, and I think
awesome.
they're all gonna have to stay here.
Yeah,
Nothing's better than just like a normal subok with the browns and the reds, and then the white in the saddles, and the perfect saddles they have.
Snakes. Yeah, they're really cool. I love them.
The babies are super defensive too. They're pretty funny.
Yeah.
But obviously they're in a genus with Baja rat snakes that Bill Hughes works with, and they have different. They're in the same genus, but they have different chromosome counts, which is kind of weird for animals to be in the same genus. Would that
you see
possibly
that across
cause
well.
like a?
Yeah, you see that across boas. Yeah, so within the boas, you'll see that occurring as well. Which is why some of the hybrids work and some of the hybrids don't work, depending on the crossing as well. Yeah. So even
with that,
even some of the insects that we work with, you can have species that are somewhat closely related. Like we've sequenced we do a lot of work with bed bugs, related to indoor kind of evolution or evolution in the indoor environment. And one species that's really common has 15 chromosomes, 13 chromosomes and two sex chromosomes. And a closely related species has 16, which is 14 chromosomes and two sex chromosomes. And we can, through genome sequencing, we can show you which chromosomes split to form these other, these two. So that can occur as well. Yeah.
Okay. I always just assumed that because of that they should have just been separated already, but obviously that might not be fully correct.
Yeah. No. You can find different numbers of chromosomes across different genus, different genera.
Wow, that's kind of cool.
Yeah,
I never knew that.
Chromosome evolution's it's voodoo. Why some chromosomes split? Why some fuse? It's pretty fascinating stuff.
Which boas have the different chromosome counts in the same genus? Is it just like Imperator in that, or
through Epicrates and Chalabothris?
Oh,
I believe
okay.
there are ones that will split between 33 and 34, 31, and so on. Yeah.
that would make sense? They like because Kylobothris especially like they've been separated.
Yeah.
Some clades are millions of years separated from the
That's
other.
exactly it. Yeah, that's exactly it.
Yeah.
Yeah.
That West Indian Boas book is an awesome read. It's
That
like
is,
a scientific textbook, but it's easy to read. But it's also like you gotta lock in to read it at the same time. It's like not too scientific, but it's still a textbook.
Yeah, I've got it sitting on my desk in my office because it's Graham Reynolds wrote that. Graham's a friend of mine, and he writes really really good papers. So as soon as that came out, I picked it up. So it looks like a really cool book. I don't keep any of those kind of lineages. I don't keep any Chalabothris, but they're pretty cool. So I look forward to really sitting down at some point when I'm free to really get into that. I just bought another one recently. I got one on northern pine snakes, which just came out, and it's really cool. I don't keep those either, but it looks like a really cool book.
You should run the Kentucky pines compared to the New Jerseys and see if you can get those their own subspecies.
Yeah, trust me, we're doing a whole bunch of work on scrub pythons, and that's enough species splitting.
Oh yeah, you're gonna. It's gonna be worse than the green tree split.
No, it's gonna be quite comparable.
Oh, okay.
Yeah, so
because they're.
I was gonna say, isn't there
and Ryan Young for quite a while?
I was gonna say, isn't there green trees pretty much everywhere where there's scrubs, and it's kind of usually the species split tend to follow the same as everything else. So I feel like it'd be very similar.
Yeah, you can look across Papua New Guinea and the islands around it, and you can see the same story with birds. You can see the same story with lizards. You can see the same story with butterflies, with beetles, with all these different things. They all follow the same pattern. And Nick would and probably has told you all about the biogeography and the paleogeography of Indonesia, and the story's pretty cool. Like we've done trips out to Hawaii to look at samples of scrub pythons together, and whenever you sit down with Nick, and he'll show you on a map how this island formed by things smashing together, and it makes you realize why some lineages could be close together, but they're evolutionarily really old and quite distant. So the scrub pythons are really interesting, and we're finishing up that kind of work. So I just finished up the last sequencing that we're going to be doing for those, and hopefully we'll get that paper out later this year, or maybe early next year.
Are you a co-author on that with Daniel?
Daniel's not part of it now.
Yeah. Oh, I thought he was the one redoing. Oh, okay, that's interesting.
No, we've been doing this for about 4 years.
Okay.
And Nick, myself, and Ryan Young. Yeah. So
so this is a book, or is this just a paper?
Just a paper.
Oh, okay.
Yeah. So it's
wow.
Yeah, we've done a whole bunch of work on genome and speciation in scrub pythons.
I was gonna say, is the main goal to speciate and properly separate scrub pythons? Yeah. Okay.
And we've got some really cool stuff that people won't expect.
That's why he always talks. Sorry, I was gonna say that's why he always talks about the undescribed species that he knows exists.
Yeah, and there's even more than that we're not going to describe in this paper. So there's the stuff out by the Bismarcks, and there's some really weird things there. We spent time. Nick and Ryan and I flew over to Hawaii just before COVID, maybe.
Yeah,
And I
I
think it
think
was
you
just before
mentioned that.
COVID, and we spent like 8 hours at the California Academy of Sciences in San Francisco, and then we got a flight to Hawaii, and we spent 40 hours in Hawaii at the Bishop Museum, and we just worked on scrub pythons for that entire trip, and we found these like 4 foot long scrub pythons from the Bismarcks that were jet black and clearly had just laid eggs, and they were adults with big heads and morphologically very different than what you see in other scrub pythons and so on. So I think what we're going to describe will be um, pretty straightforward, and then they'll be worked on the line from there once we get fresh samples to be able to do the Bismarcks and stuff like that there.
Just by curiosity, so you guys are going to describe them mainly on chromosomes or on
Oh, no, no,
percentage
no, whole
difference?
genome,
No. The-
whole genome evolution.
Okay, so when you do genome evolution, are you considering like, well, if this animal is 5% different than this scrub, they're a different species? So what's your marker? What's your number? Because I know like if I go to inland, they were 4.9%. That's why they didn't get elevated to a species, right? Which is bullshit. So just by curiosity, where are you guys lining up with the that magical number? And I know it's different for everybody and who does it, but I'm just curious: is your number 5% too, or is it different? No, not at all.
It'll be based on, so not only do we use scrub pythons, but we add in other species, and from that you can use an estimate or an average between other species. But we've done work on on, on cockroaches recently, where we've shown species that have evolved about 2,000 years ago, and they are about 1.8% divergent from their closest relative, and yet they're different species. You can see stuff. The classic work that came up with 5%, was done on butterflies. It's different on every lineage we look at. So we just got to see how samples cluster together and how long the branch lengths are in the phylogenetic tree between those to determine whether the species or subspecies. I'm not a huge fan of subspecies, but I'm okay with it. Like I think the green tree python paper was good, but I think it shot itself in the foot by making subspecies. The subspecies are clearly on their own evolutionary trajectories, and they are speciating to the point where they will be full species, and they should have been named as full species. So we'll probably be a bit more conservative with that and be species related, not subspecies. But the number it'll be dependent on, realistically, in the end, when we look at whole genome evolution, both nuclear and mitochondrial, to determine kind of what numbers we're coming up with. But based on the phylogenetic trees that we're producing so far, they're very, very clear. They're not short branch lengths. They're very clearly speciated groups, which is comparable to what you see in the work on green trees and comparable to what you see in the work on lizards, birds, insects across that range.
Wow, that's pretty cool. I remember, I think it was probably on Cush's Corner that Cush and Ryan Young were talking about how Ryan was busting Steven's balls about. I think it sounded like he has most of them, but he might not have bred all of them, kind of thing. But I think that was just kind of neat how it's like, oh yeah, there's only this in the hobby, but then oh no, those are actually this and that and whatnot.
Yeah. I think Nick. I don't know if it was on your show or another one. Like Nick did let one of the new names slip, and
he definitely did.
Was it our show?
He did on one of our shows. I was going to say, if you patch every show together, I don't think he slipped the same word on each of them. So I think if you actually listen to all of them, you can eventually figure it out. Yeah, he did slip. I heard him slip.
Yeah, that's cool. It's fine. So it's we did try to bring in a group in Australia to be involved in the work, and it didn't pan out.
And they're all children.
They never came through with what they promised, and so therefore we just decided to work on our own. So.
Yeah. But what's your thoughts on that children paper that is
Yeah, I need to go. I
They're all
haven't read it in
children.
that long. Yeah, I keep. So I didn't even. Yeah, I did mention I keep a lot of children. So yeah, it's. I need to go back to that paper. It's been that long since I've read it, but yeah, it didn't make a lot of sense to me the way it was resolved in the
Yeah.
end.
We're not even going to talk about the new species we could have described
I
and
know. Yeah,
got
they never
got
talk about
bonus
pygmy
points for.
bandits. Yeah, the pygmy bandit was an obvious one to talk about, and then they talk about the one on Papua New Guinea, was
PNG.
it
Yeah.
Papuensis, and it's the differentiation of that is so minor. It's like the equivalent between Darwin's and Papuan carpets, which is they're the same species.
Yeah. Exactly.
So
Or Viridis, right?
right,
Like
right.
they're
So
all.
it didn't make a lot of sense to me. I should go back and read that paper again because it's been a while since that came out,
Yeah.
and I was immersed in other stuff at the time, so I never sat down and read that to the level that I should have. And then it just kind of
Yeah.
slipped by the wayside, And now that I keep a lot of Antaresia, I really should sit down and go over it again.
Yeah.
Um,
It's kind of
yeah,
annoying that some hobbyists now are like, "Oh yeah, they're all children right now." It's like, no, no, don't even go there. Don't
yes,
don't even breed your
you know,
Stimsons to a
but it's
children eye.
yeah,
That'd be
definitely not.
stupid.
Yeah, but I'm sure sadly that's happened over time.
Yeah.
People have done that because for the odd hobbyist that doesn't know the difference between a Stimson's and a children's, then they'll just
Yeah.
interbred them. And of
Or,
course, I'm
or
sure there's areas where it does occur in the wild, but you get intergrade zones. But we should
exactly.
still try
But
to we
that's
should still
different.
try to maintain our. We can't go back again, right? Once we go into the dark side, we can't come out of the dark side. It's been
Yeah.
hybridized, and that's it. I love these ideas. These people that can turn around and go, yeah, but if I breed it back for 3 generations, then it's back to Sigma. And like, no, that's not the way it works. you know.
No.
Yeah.
It'd be cool for somebody to sequence the escarpment childrens because they look so different. Like
Yeah.
I could see that being more of like when Iper and that talk about, well, they're all the same snake, just different paint job kind of thing.
Yeah. Yeah.
Which is fair because he has seen them all and seen them all throughout their range.
Mhm.
So yeah, that's a fair comment for some of it.
Yeah, if we can get samples, then we would do it. The
Yeah,
problem is it's not easy. It's not easy to get samples.
yeah,
So it's yeah.
because they're in the national park too for the most part, right? I don't know what their range is for the escarpments, but
Yeah,
obviously some of them are in the
difficult.
national
Yeah, it's just
park.
difficult to get tissue sent from Australia to CITES paperwork
That
and
too.
Australia being Australia and stuff like that. There, it's just a bit of a pain. Realistically, I could probably fly over there and do the work there. Sequence samples there, and then just bring back the genomic data with me to analyze it. But it's again, who's going to fund that? I'm not. There's no money in that research. There's no money to fund
Sounds
that
like
research.
time and money.
Yeah, it's
Yeah,
great if you've got that spare change sitting there and the odd 25 or 30,000 to be able to do all the sequencing, but
yeah,
it's sadly that's a question that's going to take a while. It will be an Australian group that addresses it, and I could probably guess the group that will do it. We'll just see what happens in time.
yeah. Hopefully they don't mess it up.
We'll see.
You referred to pygmy bandits. Just by curiosity, do you consider pygmy bandits and pygmies two completely different species?
Yes. Yeah.
Yeah.
Okay.
Okay, because I don't think they're
they're
separated
like-
right now, but they're not
Yeah,
separated, aren't they?
Well, no, pygmy bandits
pygmy
are
bandits
just
aren't even
a
described.
non-described.
Yeah.
They're just a
Pygmy bandits are
non-described.
described. Yeah.
Okay.
Pygmy bandits are they're like somewhere in between a children's and a pygmy. They're tiny, they're banded. They look like a children's python more in structure than a pig. Pygmy pythons are, they got a bit of girth to them, whereas a children's python is more slender and so on. So they're much more children's python-esque, but they're tiny. Yeah, they're really cool.
And they're in the spotted python range kind of thing too.
Yeah, I believe so. Yeah.
Yeah.
They're neat.
Yeah, I liked it.
Yeah, I like them a lot. I got into Antaresia 2 or 3 years ago, maybe 3 years ago, maybe 4 years ago, maybe longer than that. But they're a lot of fun. Yeah, Ryan Young sent me down a bunch of different spotted pythons, and then I got into the children's pythons, and my buddy Zach Loafman sent me a pair of spotted and children's that they didn't want anymore because they were too evil for the students that they were working with. And then I just got immersed into that whole group, and I really like that group. They're a pain in the ass to get feeding, but they're a lot of fun.
The spotteds were a pain in the ass?
Children's would be the pain in the ass. Some of them take off really well, just with defrost pink mice, and you split the face and they go for it. And other ones you gotta tail them for a bit, and then they get going and they switch over. But in every clutch I produce, about 50% feed straight away, and 50% are just a pain in the ass. They take a few months to get going.
Yeah, because Dominic, you used to work with spotteds and children's, Dominic?
Not children's. I did spotteds for multiple years. Mine were as evil as it gets. But every baby they all ate first shot, no problems of each clutch.
Yeah.
So I
the
never
spotters
had
I
any
a
issue.
problem with. Yeah, my spotters are always straightforward to get
Yeah.
going. It's just the children's are 50-50.
Interesting. So spotteds, no problems. Pygmies are the worst of the worst to get feeding.
Oh, yeah.
They're about as bad, but I've never actually owned a pygmy bandit. I've considered buying them a couple of times now, but I honestly thought it was just like a different locality of pygmies, to be honest.
No, they're separate. They're on their own. They're really cool.
So now that you're saying that, I feel like I need to get them because I love pygmies. They're one of those animals that I love. Like I've learned to love them, and the spotteds I've kept for like 6-7 years, and then eventually I parted with them.
Yeah.
So, but the pygmy bandits, I bet you I'd like the Stimsons too.
And you can get them a lot easier than here, so you could probably import from Paul.
Yeah, we can.
Really easy,
that's the list we see them on.
You just need a thicker wallet to get them.
Oh, really? Yeah.
They're not cheap.
But yeah, Paul's a great guy. I've stayed with Paul a couple of times back 25 years ago back in the UK. I sold him a bunch of ball pythons and stuff, and when he was getting into balls. And he's a great guy. He does some really cool stuff. And sadly, getting them to the US is not easy through the nonsense of Fish and Wildlife now. But
yeah, well, it's hard to get stuff out of the US.
People have brought
too.
them in. I think Justin Julander brought some in recently. So they can come in, but it depends on what paperwork they come in on.
Yeah.
Yeah, getting things from him is pretty simple, actually. There's zero issue, but for us, same thing with the US. Like anything from US to Canada, I think we say it almost every show. Just can't be done. It's a pain in the ass.
Yeah.
Like even stuff that should be allowed out, just they're like, oh, well, you don't have proof that it's been in the country for 3 generations, so no, we're not going to approve the paperwork. Like,
yeah, that's it.
You guys let this out 8 years ago.
Like, and it's amazing. It's whenever you have paperwork that shows a record of it being imported or exported, and yet it's whoever's working that day. It could be a real pain in the ass.
Yeah. It's a brown snake that nobody wants. Like,
exactly. Yeah, it's just a pain.
Yeah, like the Bahama boas I have, they're from Jeff Murray.
Mm-hmm.
They were allowed out then, and
they were.
Yeah. A guy in Calgary imported 6 up back in 2019. Okay. So I got 3 of those animals, and then Cody Joe has the other pair of the silvers. Actually, he sent me a picture this week of them locked up finally. Because his pair of Nassaus and my pair of Nassaus do not like each other for whatever reason.
Oh, really?
Both of them. Okay. Like they're 10 years old now, and you go to pair them up, and they just run from each other. Both of them run from each other. It's weird. But then there's pictures of mine being locked up too in the past when my buddy had them before me. Mm-hmm. But I don't know. She
not old.
No, I'm not worried about that. It's just. Yeah. It's just funny how like they just won't just like chill out and like I walk in the room and they're locked up. I've haven't seen them locked up, but
people don't think about snakes having personalities and having choice.
Oh yeah,
but they very much do. There's a lot of mate sexual selection and so on, and that goes on. It's not just the case of put 2 together and you'll get offspring. I see the tree bows a lot. I can put different pairs of tree bows together, and some of them lock up immediately, and some of them just sit at the opposite end of cages. They don't go near each other.
Yeah, that's why I got a 2014 brown line Bahama male from Cody Joe that produced the 1 litter 4 years ago that I have an offspring of. Mm-hmm. And the female sadly passed away last year, so I'm like, well, maybe I combat them or throw the 1 male in and then quickly pull them and then throw the other male in or something. And
you got to
try to.
Yeah, absolutely.
Although that brown line is definitely like a different snake than the Nassau.
Really?
Like the head structure is like just almost more like a scrub python head. like not huge, but like the color's different.
Mm-hmm.
They have like kind of a similar stripe here on the eye,
Right.
but it's like a gold. Definitely has like the gold head that Jeff talks about,
Mm-hmm.
and the facial structure itself is a lot bigger. The Nassau boas seem to have small heads on them.
Oh,
Like the
okay.
female
Yeah.
has kind of a bigger head, but
Yeah.
the male that's for the female, he definitely has like a smaller head. So it's kind of odd how it's like, yeah, I can see why Jeff wants people to keep these separate. So
Yeah.
maybe
Because
Michael Saina will breed
yeah,
some of those.
because again, we don't really know a lot about the true phylogenetics of them. Nobody's really ran a lot of these samples in the localities, so it's quite possible you could have species level splits occurring between these, and we just don't know.
Oh yeah,
And again, it comes back to my comment earlier on. We can't go backwards. Once you cross them, you've crossed them. And that's it.
yeah.
Yeah.
Like I know the brown line. I wouldn't be surprised if they were some sort of bimini boa instead of a strigollatus.
Oh yeah. Yeah.
Right? Because you notice the biminis will have the kind of like that gold head on them, and they're a bit bigger, it seems. But that's I don't know. The Bahamas are just so fascinating because there's 6 or 8 subspecies, but then even within those subspecies, they're on separate islands as well. So like each island
Yeah.
is probably its own. Like there's obviously within all of that, there's a bit of gene flow from hurricanes and whatnot, but not. It's
Probably not a
still
lot.
a central
Yeah.
population
Yeah.
of animals, which is super fascinating.
Yeah, it kind of it saddens me whenever I look at that group, because you can go back and you can find pictures in captivity of a lot of those different islands, and then now they're gone.
Oh yeah,
They've
I
disappeared.
it pains me to see the Ross and Marzak book, and it's like that was in Canada, that was in Canada. This is all Thomas Huff stuff.
That one there.
Yeah, I think I got the slightly newer one, or no, it might be the same one.
Mine's even signed by Ross and Marzak, I think. I didn't realize.
That's cool.
I was just reading it. Yes, I was looking at it yesterday. I got a couple of different copies of this here, and I opened it up and I was like, "Oh shit, it is! It's signed by Ross and Marzak." Yeah,
that's cool.
Yeah, that's a good book. Even though it's horrendously outdated and a lot of the information's incorrect, I still love that book. It's great.
Yeah, I even seen the Cat Island boa. That species of strigollatus in there is like that was in Canada. That's insane. Those aren't even in captivity anymore.
Yeah, it's
and he had.
I see the same thing with boas. Like Dominica boas, the clouded boas, the boa nebulosa.
Oh yeah.
And the boa orpheus. We're trying to work with Dominica and St. Lucia to bring boas in from the wild, to set up a breeding group and to do some genomic work on them, just like we did with Trinidad's. It's taking a little bit more work, but we're hoping to be able to do that, which would be awesome.
Are they at least communicating with you guys?
Yeah.
Or?
Yeah.
Okay.
Yeah. So just
you just might have to offer them some donations.
and I'm not going to do that. So it's got to come in as a proper kind of breeding project where the animal.
No, I mean more like they want some sort of like in terms of scientific.
Yeah, that's what we're
backing somehow.
Yeah, that's what we're pushing with them, right? So we'll sequence genomes, we'll create genomic stud books where we can then offer to zoos highly unrelated individuals. And then it actually, you look at those islands and they get hit by hurricanes all the time. You're losing a lot of individuals whenever that happens, and therefore we want to create some kind of genomic arc where if you need to send animals back to islands, you can, and so on, because they're not inbred. So we're working on various projects with them. So we'll see what happens. It'd be awesome to bring in the St. Lucia boas and to bring in Nebulosa, because in the UK, whenever I was there 25 years ago, a friend of mine who was a big boa locality guy, he had Nebulosa and he had St. Lucian boas, the Orpheus, and they were kind of cool. But I didn't realize that 10 years later, 15 years later, there was going to be none in captivity. It's just like, oh, there's that boa nebulosa. It's kind of cloudy and dark and aggressive. You always just walk past them because they were so freaking aggressive. And now, if I walked into somebody's collection and saw those, you'd kind of wet yourself and seeing nebulosa.
You'd be like, where the hell did he get that?
So it's looking back in a lot of these books and magazines. And
Jeff has a pair of
Nebuloso.
Nebuloso, right?
Yeah, I've known that for years,
Yeah.
and he let it slip on a show that I was
Yeah.
recording.
It kind
Um,
of seemed like he kind of chilled out that episode, which is pretty neat.
and I was shocked. Like I literally was shocked that he turned around and admitted that he had them. Unfortunately, his mail's not that great in terms of
Yeah.
a breeder,
You,
but
you send him down a male imperator yet?
he's got something there
No.
to be able to try it this year.
Oh, okay.
So I hope he does, because I was actually thinking about that today, and I was talking to some colleagues in the vet school at my university about using potentially artificial insemination. So we'll see more about what can be done with Jeff's animals, and again, you lose the female, you lose the male, and you've lost a project, which would
Yep.
just really suck. So,
That's kind of the same state as the Exul project right now, I believe.
Yeah.
Because that same guy still has the 3 or 4 animals that were bred 10, 15 years ago now.
Yeah.
So
Those
are like at least they bred then, but still.
Right. And the animals can be old whenever they're like, look at Dunn's pythons, right? So the Dunn's pythons that whenever Ryan and Nick KJ bred them, whatever it was, 5-6 years ago, those animals were really old. So they still can be, boas can be old. I've had boas that were in their 40s. I've got boas here that produce litters into their 20s, no problem. So they should still be able to reproduce, but we'll see. You have to have the right people doing it, and they have to be in the right condition and so on. So, but if they're not breeding, and we can start using technology such as artificial insemination, then that's the way forward. And that technology has been advancing quite a lot with reptiles. So I hope that for some of those kind of rare projects, there might be a desire to do it, which would be kind of cool. But again, it's there has to be investment to be able to have people willing to do it. Either a cool vet that's willing to invest their own time, or somebody that's going to invest and pay the vet the money. The scientists are going to do it.
Yeah, that's the tough part.
Yeah.
It's frustrating because up here in Canada, I think there's like a small group of really old guys, or I don't know if they're really old, but old school guys out in Ontario that have some of this.
Yeah.
Cool, like they probably have subflavus and inornatus that they've had for 30 years.
Yeah.
And apparently, like one guy was able to find somebody that had inornatus still, and the guy's just like, they're too old to breed. I'm not gonna sell them or breed them. And it's like they're only 20 years old.
Yeah.
Like,
and all right.
but
so,
unless
so
you're neglecting it,
they're just going to
it should be fine.
You lose it. Yeah, absolutely. Yeah. Put them through a cycling phase for a couple of years, especially if they've been in a collection for that long. They're cycled to the room that they're in, so there should be an opportunity to give it a go.
There's CITES records. Somebody brought in like 40 Puerto Ricans into Canada legally in the early 2000s. They should be everywhere. And plus, what Thomas Huff was doing in the 70s and 80s, breeding the Jamaicans and that. Like, I know of one Jamaican pair in Canada.
It's
I don't obviously like there should be more than that because those babies came from somewhere.
Yeah,
but still,
whether they're sitting in collections and they're just being maintained, and these people have had them for 20 years and they're not big into social media and all that kind of stuff, I can totally understand that.
Yeah.
But it's sad to see those animals that have the potential to repopulate. Because look at Dunn's pythons now. Like shit, I had the chance to buy Dunn's pythons 25 years ago back in the UK, and we didn't because they were just ugly brown snakes. And then they disappeared. And then Ryan gave me a male 1 year, and then a couple of years later gave me a female, and I bred them this year. I bred them this year, and I had people biting my hands off to get those animals because they really wanted them for a lot of money, and I just think like shit, if they hadn't have done that, then that species was lost essentially to captivity, which is just like a lot of these Chiloboptera. It's just like a whole bunch of Corrales, the lineages and so on for Corrales. Like Trinidad tree boas were in captivity for a long time, and Cook's tree boas were in captivity for a long period of time. The problem is they're a pain in the ass to get feeding as babies, and whenever things are a pain in the ass to get feeding as babies, people lose the interest in wanting to breed them, and therefore we lose those lineages. Just so things like Nebulosa, I'm sure, are probably lizard feeders as babies, and they're going to be harder to get going. And and they've got specific requirements for breeding that are totally different than a Colombian or a Nicaraguan boa and so on. And
Yeah.
once you get into those little species that are more finicky to get going, then they just kind of disappear in the hobby. You need people that are really invested in it.
Is there any Kylobothra stuff that you would potentially work with if it became available to you, or are you just like staying in the lane that you're kind of doing right now?
I don't know. Tom Crutchfield offered me Jamaicans a whole bunch of times, and I just don't have space.
Big musky, bitey snake.
Yeah, I think that's part of it. They look beautiful. Like whenever I was in Oklahoma and Tulsa, the Tulsa Zoo had a really nice Jamaican boa, maybe a pair. They were beautiful. But for me, it's because of the inability to sell them outside of your own state, other than gifting them. And then, they're getting them feeding as lizard feeders and stuff like that there. I've got my time for that is invested in my Ruschenbergeri and stuff like that there, So, but I remember seeing Jeff and Ryan maybe 18 or 19 years ago, maybe 16 years ago, and they had a table at Daytona, and they had a bunch of little unusual Chilobothra species.
Oh yeah,
and I thought they were really cool.
The Ford-Eye are neat.
They were
cool.
really cool. Yeah, really neat. And
I was offered some when I tried to import the strigs that I was trying to import that didn't work out with U.S. Fish and Wildlife, but.
Right.
I was like, I was like, oh, I'll just keep it simple and just get the Eleuthera, which I wish I got because they'd be cool. But
yeah, I think if I had more space, I would.
If Jeff ever offers you Bahamas, you should do it because Bahamas they're not musky. I've never been musked by any of my Bahamas,
but they're slightly
and
bigger,
they're only just they're like males are 5 to 6 feet, but they're super thin.
Yeah, okay.
Like they're like half the size of a female, and then the females are about
they're
like a rainbow boa size.
Yeah. Maybe one day we'll see.
They're fun. They're cool. They all look different.
My space is fully absorbed. I don't think I have the ability to get anything else. Thinking about it right now, my one room turned into 2, and 2 turned into 3. And my wife is not happy about any of that. So, with my Timor pythons, Ryan Young gave me a pair of captive Timor pythons a year ago, and they grow fast. So I'm going to have to get some 6-foot enclosures for them, which wasn't on the horizon for that room. It was meant to be a storage room, and that's a storage room plus Timor pythons. So I don't think I got the space for other snakes. As much as I'd love to have a whole bunch more, or the time. It's a lot of work going through everything I've got, especially with a full-time job and young kids and everything else.
You got to move things out to move things back in.
That's exactly right. And the problem is that I don't see anything that I want to move out, So I keep thinking about that. I'm like, well, what if I sell 6 of these or 8 of those? And I'm like, yeah, well, if I do that, what if I want to get them again? Or, what if I, so... Yeah, I think I'm at my capacity for species and for space.
So
without making it your full-time job.
I don't want to do that. I don't want to make my hobby my job. Yeah.
So you went into something I found a bit of interesting. So a couple of times you talked about duns and other species where basically we had 2 last animals and kind of repopulated, if we can say, the captive market, right? Yeah. So it's happened a couple times with probably a couple of species actually, and most of the times from what I can tell, it seems to be that the animals are pretty resilient, do pretty well with a very, very restricted gene pool, right? I'm wondering if you could explain why inbreeding seems to be a very negligible problem in reptiles, specifically worse or better in certain species, but for example, most mammals are, I don't want to say like higher beings, but animals that seem to be more, I don't know, not more evolved, but more complicated. I don't know the best way of saying it here. Mm-hmm. It seems to be, inbreeding seems to be more and more an issue. When you keep going down the line, like for example, cockroaches, you could have inbreeding for hundreds of generations and have almost no issues. So reptiles tend to be able to I'd say you probably could inbreed 5, 6 generation and often not even have a problem. So why? I might be exaggerating, but I don't know if you want to
I
go
think it
down the.
differs. Yeah,
Okay,
I
go
think
for
it
it.
differs between lineages, and I don't think we know a lot realistically about it, right? So snake keeping, snake breeding is actually still a really young hobby. Um, 30 years ago, whenever I got into it, there wasn't a lot of people really breeding snakes in Ireland and stuff, right? It was still very common to get wild caught. Um, and 30 years, that's 10 generations for most species or less. That's not a lot of time. Um, I think we over exaggerate how resilient snakes are or reptiles are to inbreeding. I think we don't really truly know, because if you look at some things like hognose snakes, they are not resilient to inbreeding at all. You can go 3 generations and start getting eye deformities and tail deformities and so on. Um, we look at some boas, however, and they're really resilient to it, right? So the K cocker, cocker's K, lagoon K, these little island boas are really resilient to it. But the difference is that hognose snakes have had a massive population size for a long period of time. Yeah, it's contracted now due to habitat fragmentation, but there's always been a lot of genetic diversity in those populations. In contrast, an island boa that has been isolated on an island for tens of thousands of years, through selection and through the elimination of individuals that weren't fit in that population, you've reduced the gene pool, you've reduced the population size, but what's there fits perfectly into that island. It does everything it should do. So inbreeding works because of the deleterious alleles, the alleles of genes that are detrimental that would be in populations normally have been eliminated, and therefore they can inbreed and they do fine. You inbreed some of those, right? So my buddy Scott, who brought in those Cocker K and Lagoon K originally, when I look at his animals now, 20 years later, you find some really unusual patterns evolving, in those, 6, 7, 8 generations down the line. There's evidence of some jaw facial issues or some evidence of eye issues. So it still does impact them, but just not to the same extent. But it totally depends on whether the species that we're working with came from a very large gene pool or came from a very small gene pool that was selected for over time. Because not all of them do really well. A lot of things crash very quickly, and we've sequenced a whole bunch of genomes, and we find that a lot of things in captivity, like ball pythons, the individuals are really genetically depauperate. There's not a lot of diversity in those in individuals. As a species, there's lots of diversity, but within lineages, there's not, because people are buying a pair and then pairing a pair and then breeding a pair, and there's not necessarily a massive amount of outbreeding going on because they're getting their desert ghost het exanthic het clown and they're breeding it to a clown, and yet they don't really, they think it's outbreeding. And I'm like, well, the clown came from somewhere. It came from a single introduction generally from Dave and Tracy Barker, right? There's a lot of inbreeding still there. It can be lineage specific as well. So you can find some lineage that work really well because they purge those deleterious alleles, and you can find some lineages that just crash really quickly because they still have deleterious alleles. And sometimes if we outbreed those and we cross these lineages, it actually works out more detrimental because we're introducing deleterious alleles from one lineage into the other. They can then cause problems down the line. Inbreeding is a really complex story. It can work out really well for some species, and it can work out really badly for others. In mammals, it turns out to be really bad in most cases, for most mammalian species that we know of, and same with birds. The reptiles seem to be a bit more resilient to it. Insects tend to be very resilient to it.
It's
Yeah,
interesting. Sorry, go ahead, Mike.
yeah, I find it really interesting with like the island species stuff, how they seem to have gotten rid of their deleterious genes and whatnot due to
Yeah.
a long time. But have you heard of the Kentucky Pine story with the Phil Peak episode and all that?
No, I
How
haven't. I
they're
haven't gone back to that one.
okay.
Yeah.
Yeah, that is a good one. I remember seeing Phil's original comment on one post about him, and he just it was like like 4 paragraph pages on a Facebook comment thread about how they even got them. But there does seem to be a little bit of genetic inbreeding already with only
Yeah.
the 3 original animals from 3 different counties from Kentucky because they're
Yeah.
I I I think the inbreeding with them is bad because they're not they're like an island population, but then they're not because they probably 100 years ago had a broad range
Okay.
going down
Right.
into Tennessee and even northern Alabama,
Yeah.
where you have all those yellow animals, or Georgia,
Right.
whichever one it was. And
Yeah.
now that they're reduced into their own pockets, and then you start crossing them, they haven't worked out their deleterious genes.
Right.
And then, like, I think I've heard you or other people talk about, well, yeah, if you were to breed something into a Clocker or a Hog Island, it might actually introduce deleterious genes so
Yeah.
that they're not as strong anymore.
Absolutely.
So I think that might be like a thing where the genetic diversity isn't there, and because they already started inbreeding in like not a good way in the last 100 years. That's probably why you're getting some of that.
Yeah, like we see it in indigo snakes. There's inbreeding issues in indigo snakes, inbreeding issues in hognose snakes, and so on. Yeah, so we talk about inbreeding depression, but there's also outbreeding depression where you can cross lineages and actually have a significantly detrimental effect as well.
So just to go back, let's say to the hognose snakes, they're completely opposite of an island species, right? Where there's tons of diversity. I feel like if you were starting to get some inbreeding, if you breed it out to unrelated animals, you might get outbreeding depression, but you might also fix a lot of the issues, right?
Right.
But with so much selection, wouldn't you just be able to easily fix those problems? Because there's so many different lineage. No?
Yeah, you'd think so, but I think a lot of those lineages have been derived from a very small, shallow gene pool, um, and I think the problem is that people inbreed and inbreed and inbreed, and very rapidly collapse the genetic diversity in an individual. That when you then go to outbreed, it can actually just generate outbreeding depression problems because you're breaking these, you're introducing gene complexes that are deleterious, you're breaking gene complexes that are good. It can actually be a really, really complex story whenever you look at it at a genome scale. And again, that's a really interesting area for research. We're actually doing that research with insects, where we're creating these inbred lineages, and we're putting them through generations and generations of inbreeding, and then we're outbreeding to see how those genomes react, to which regions of the genomes are actually are recombining, which genomes are staying homozygous, which genomes are varying, or which regions of the genome are varying. It's actually a really complex story, and it's one that we're finding out is almost species specific and in some cases lineage specific. With hognose snakes, it's because they went from very large gene pools to very collapsed gene pools very quickly. Whenever we bring animals in from captivity, we're bringing in a few individuals, and they become the founders of our population. So you get what's known as a founder effect starting off with it, right? We're starting off with a shallow gene pool that we then make even more shallow over a handful of generations. In some cases it works, and in some cases it doesn't. We also got to remember that with every generation, per billion bases of genetic data, we get about 40 new mutations. So new mutations are arising all the time, and they can be detrimental or they can be neutral. They can have no effect, but we can be adding mutations in that are also problematic down the line. Which is why we find some things like, I know that with hognose snakes, there's the pink pastel albino, which has has been problematic, and they can outbreed it and it fixes it a bit better. But there's been inbreeding that's caused that because they try to fix traits. Anytime you get a new mutation, the first thing you do is inbreed the hell out of it because you want to try and fix that mutation, right? And then what problem is that people then don't outbreed to then try and increase diversity into those lines. Like I've always said that the thing that amazes me about the ball python hobby is that they've ignored one of the greatest sources of genetic diversity for years, which are all those wild captive farm females that are $10 a piece. And to buy a bunch of those to breed their 12 gene animals into to increase genetic diversity, then to be able to cross back from, is a massive.
Do you think they did that originally though?
Probably not.
Because didn't a lot of them buy up tons of normals
Oh yeah,
back when the
early
genes
on,
first
yeah, yeah,
started, and then they started.
absolutely. Early on,
Could
yeah,
could
they
that
got
be
their
a
one
positive for
albino.
why they haven't? Yeah.
Well, they got their albino and they bred it to 50 wild caught. Yeah, but after 10 years, they stopped bringing in wild caught for that purpose. Right, they stopped using that. I would imagine if you go into any of the main ball python breeders and look through the racks, you'll not find a single female wild type ball python that is wild type with no known hets. And I think if they had them, they got rid of them a long, long time ago.
No point in keeping those.
No, that's right. And yet they're an incredible source of genetic diversity.
So if we go back to the inbreeding, so if you take like an example of like I hear a lot of people talking about like breeding rats and breeding mice and all that, and they're going to be like inbreeding the crap out of them, and they will not bring any other rats from anywhere
Right.
else, because once they have a population that breeds and continuously inbreeds back to its children repetitively, and you're talking about, sometimes they'll talk about 30, 40, 50 generations with no issues.
Yes.
You bring in new stock, and then all of a sudden everything dies, right? Or it causes this
Yeah.
gigantic thing. So if I go to now reptiles, if I'm understanding kind of why the way you're describing it is that island species have kind of already, either they survived or they didn't, right? So if they survived, they're kind of like the
Exactly.
rats, right, where everything is good. So
Yeah.
then if you take an example of, for example, like Papuan carpets, where granite came in and everybody bred the crap out of granites and inbred, and all of a sudden we had all these issues.
Yeah.
Now, they were outcrossed, and outcrossing fixed everything. Is that just because it happens that the outcrossing had no deleterious effects in them or genes in
Yes.
them? And
It means that if they did have deleterious alleles, they were different than those that were within that line, right? So through all of that inbreeding, you got the issues with the original granites. Outbreeding them, or breeding them to those wild lines, just it adds a flush of genetic diversity to the offspring. And there's still going to be those deleterious alleles, but they're in the heterozygous form, they're not expressed. Now, what will happen is if you take a bunch of those and start inbreeding them again, then you're going to get those, very likely you're going to find those problems arising again. It's just that we've been able to outbreed those lines, and we were in the early stages of crossing them back together again to produce the homozygous form, that we're not seeing all of those deleterious alleles expressed. But if we inbreed and inbreed and inbreed for another 3, 4, 5 generations, we'll be back to square one, as we were whenever the first problems arose.
so now, I honestly, like, I've bred a lot of granites now, and I've never ever had any issues. But most of mine came from hets that were outbred, outcrossed, and all that.
Yeah.
Now I can breed granite to granite, and I still have no issues.
Yeah,
Now,
that's
is the idea that because they now came from half their genetic was, let's say, a wild caught and so on, now that because you've brought all this different genetic diversity, it shouldn't be an issue unless I would breed brother to sister 3 or 4 generations again, I'd probably end up in the same place.
Potentially, yeah. Yeah. So think about it, right? You've started off with a genome that had deleterious alleles in it, right? That were problematic. And if you bred sibling together, then they're expressing those bad alleles together, and you got problems. So instead of doing that, you breed one to a wild type and you outcross it. So the offspring, some of them have those deleterious alleles, some of them don't, and in different combinations. Whenever you breed those back together again, the likelihood that they've got the same combinations that are then going to create the problem is reduced. So you're diluting the whole number of alleles that are deleterious. You're removing some from the population, and they're still there. Some of them are still there, some of them are gone. And it depends on what way you cross individuals that's then going to determine whether they're expressed or not. What you're looking for is just that granite, right, is what you hope to find. And what you're hoping to not get are all those other ones that cause the problems. So the outbreeding dilutes those deleterious alleles in the genome and reduces the likelihood that you're going to see them expressed in the offspring. And if you can outbreed a couple of generations, then you can, as long as those alleles are not really tied to that gene. So what can happen is you get a process called genetic hitchhiking. You can have, say you got a chromosome, and at this point in the chromosome, you've got your gene for granite, right? Or your mutation that results in granite. If at the other end of the chromosome you had a mutation that caused tail kinking, the likelihood is that across that, at some point in time, the chromosomes are going to cross over due to recombination. You're going to lose that. You're going to break that pair, right? Through outbreeding, you're going to retain hopefully just one or the other. However, the problem is that if that tail kicking gene is really close to that granite one, then the likelihood of recombination occurring where you split that pairing is really, really slim. Right, so you get that process called genetic hitchhiking, which means that the bad gene or a gene just hitchhikes with another one, and they're always inherited together, or they're very likely to be inherited together. That can be one reason that you get these problems. The other is that, I mentioned about a lot of these mutations are from pathways that are really long. One gene turns on another and so on. If that's in a pathway that is linked, you'll never lose it. It's like with spider ball pythons, the pattern mutation and so on is tied to inner ear development and so on, right? So you're always going to get that problem, just like with scoria boas, just like with, I believe, the skull face and western hognose snakes. You'll never be able to uncouple those because they're part of the same genetic pathway. But if they're not, if in granite carpets, for example, I believe that was just due to a very shallow gene pool and being able to separate those deleterious those deleterious genes, the alleles of these different genes, being able to break that combination allowed you to now have those granites that are healthy.
Oh, that makes complete sense. So I love that you brought up the spider, the jag, and those are all the same genes. I think there's that same gene with the retics and other things. And I understand that you can never separate it because I think it's the melanin pathway that causes the neurological issues, right? But when you go to tail kinking, and I find that one interesting because tail kinking, it's the same gene. I think it's the petite in blood pythons. I might be wrong, which would be the same as the zebra and carpet python. I think it's in another couple of genes, but so that one is not a pathway problem. Then you're thinking it's just a hitchhiking problem.
It could be.
It could be.
It might not be. Yeah, I'm just saying that as an example. It could be. Yeah.
So I'm just wondering. So when you're talking about jags or spiders, they all have it. Yeah. When you're talking about zebras or petite,
they don't all have it.
They all do to a certain extent. Like I'll just talk about zebras and jungles. They all have a shorter tail. So their tail, if you were to always compare every single super zebra that is the homozygous form of the zebra, they always have a slightly shorter tail than a normal, like a normal jungle or a zebra just altogether. So there is, but some have kinking, some do not. So I'm trying to figure out why would that be? And if it is hitchhiking, would it be possible at some point that you could technically separate it if you happen to have the right split at the right place, or is it really completely tied together?
If it's hitchhiking, then there's a theory that there's the possibility you could separate it. But if it's part of the same pathway, it's due to gene expression patterns, which is why we see things like jag, things like spider, things like scoria that are on a climb. You've got ones that appear completely healthy, and you've got ones that spin out, and they're totally useless. And it's likely due to the gene expression, the number of copies of a certain protein that's being produced or whatever, the genes that are being expressed are at different levels depending on where they are along that climb, which is totally dependent on the combination of genes that have occurred through crossing over and so on. It's not a straightforward question and to address because we haven't seen the crosses. We've seen it with some things, right? You've seen people that say, "Oh yeah, I've crossed jag with non-jag, or one that doesn't spin and so on." But and you get completely healthy offspring. But I've also talked to my buddy Rich Eiley, who really did a lot of work with the scoria boas, and he said, "Warren, I had scorias that didn't spin and were completely healthy, and you bred it to a normal, and half the offspring were just terrible, half the offspring were fine." And obviously, it was only those that were carrying the scoria gene that had it. Those that are wild type didn't have that at all. So at that point it's likely to be a gene related pathway issue that's occurring. Otherwise, you would have wild type that would spin, or wild type that would have tail kinking and so on. So, with those, I don't believe outcrossing will ever lose those characteristics.
And they all make a dead white snake, right?
Right, yeah, with those, yeah. So I don't know, there's been enough people that have crossed ball pythons with carpets. Has anybody done a spider doing a
Yeah, they
pad?
have.
They have, yeah.
That
They
cross has
have.
been made. Yeah, there's been
Was
ball
it a dead
pythons
white
and
snake?
carpets. Oh, I don't know about that specific morph, but
It'd be funny
I
if
know
they
the
were a
cross is
lilac
made.
too.
Yeah. I'd love to see
Yeah,
it
I
in
think
nature.
they are actually. I think they are a lilac because it seems to be the same identical mutation
Yeah.
across the board from.
We need to get
Uh,
Nick to try this. I'm sure he
yeah,
might have a spider somewhere.
I hear Nick loves to create hybrids, so I'm sure it'd be a thing.
Yeah, it's gonna happen.
Science
Not
project.
except for his first breeding or two. Yeah,
Yeah.
yeah.
So I was going to say, um, is it just me, or maybe I'm wrong on this one, but it seems to be more pattern mutations tend to have more detrimental problems or tend to have more issues than actual color mutations. Because if you kind of look at it, all the most of the morphs that have problems tend to be pattern mutations, right?
Hmm.
Is that a coincidence, or is there a reason behind that?
No, I think you're right. I need to sit down, and I haven't really sat down and mapped out, but it does make a lot of sense. Because of the how melanin is distributed and how that is related to different gene pathways. It's not a simple on off for many cases. Yeah, I haven't sat and thought about that. That'd be an interesting thing to map out to see which ones. If we look at just color versus pattern, how many have issues? But I'm thinking about it with boas, motley, Aztec, scoria. They all have clear issues with their homozygous form.
I'm thinking with
Albino and
Morelia,
heterozygous.
and with Morelia, it's all also just it's pattern mutations too. I'm just wondering,
Yeah.
is there any color mutations that have linked?
Uh,
I'm purely going through it because to me
yeah, I
it
don't
seems to be
know.
really
Yeah,
pattern mutations that seems to often have that associated
yeah, yeah.
issues. And
Oh yeah, there's a
Yeah, I
bunch
think
of
so.
boomorphs, right? Like there's the Roswell laddertail,
Yeah, but that's
Celtic.
that's pattern. Yeah,
Yeah,
Celtic's pattern. Yeah, yeah, they're all pattern. Leopard doesn't have any issues, and it's pattern.
yeah.
So
well, I'm
that,
not saying that all pattern
But it's
mutations have
almost
no,
more.
issues.
no,
I'm
yeah,
saying
no,
that
yeah,
when they have issues, it
yeah,
tends to be
yeah,
pattern mutations.
yeah.
Or I'm
No,
just
you're
wondering if
right. You're right.
there's a reason for it.
It's
Mm,
due to likely just the way pigment is distributed, and
yeah,
whenever these gene pathways are occurring, what how pigment is hitchhiking along or being distributed along these different pathways is likely the issue, because what we see is just a phenotype. We're not seeing what's happening at the cellular level, right? Melanin is distributed throughout the organism as well. Not just at the external features. So it'll be related to that there. But that's yeah. The movement of pigment is very different than the change of pigment, the loss of a pigment. So it's going to tie into that there. But
is
that is
this?
interesting. It'd be interesting to look at that in colubrids as well. Do people see it in colubrids as much as they see it in boas? And I wonder, like in
I
ball
guess there's
pythons,
that
do we
one
see a lot of?
cock's eye.
Yeah,
Rob talks about that jag cock's eye kind of thing, but that's kind of not as prominent. It
and
seems like
there's a
maybe it
whole
is the
bunch of
same thing,
yeah. There's a whole bunch of
but I
pattern
don't.
mutations in ball pythons. Do we see it in ball pythons as well? I don't follow ball pythons much.
Well, I guess it's just the spider, at
Yeah,
least. But then there
yeah.
may be something else.
I'm sure there's
Oh,
a lot of them. But
hidden gene Walma, I guess too.
Right, yeah, but that's all part of that same kind of complex, right? The
Yeah,
Woma, the Woma, or
I think you're
Bengal,
talking to the
or
wrong people if you want to talk about that.
yeah,
'Cause,
absolutely. Yeah, I'm interested in mutations, and I do have two ball pythons, but they're parthenogens. They're in my basement. Um, but that's a really good question, and it'd be interesting to map out a lot of these mutations that have problems and determine whether it's color or pattern. But you're right, pattern is more likely. It makes more sense just due to how pigment is distributed and the complexity of pigment distribution, and how that's just part of a pathway that has a much greater effect on the organism.
that's awesome. I think before we close it out, maybe we should hit one of the animal related topics I wanted to hit. I had like a
sorry,
list of
Michael.
like
Pick one.
I wanted to hit like how you did Amazons and then the Rushis and then
Yeah,
your
whatever you want.
Pick
Cayennensis and
Pick one, go
in the
down it.
duns. But I think if we pick one, and then we have you on in maybe another 4 or 5 months, and we just hit straight boa stuff, that'll be a totally. Although this is awesome, like I love this episode.
Yeah, cool.
Why don't we just hit the duns to keep the Python people happy? So you got yours from Ryan, and
Yeah,
I
I got
assume
one
you
from
got
Ryan.
one from Nick.
No, both
Okay.
from Ryan. So they're two different breedings. One was from, I think one was from the first breeding that Ryan and Nick did with KJ, and Ryan sent me a male, and I had it for a year or two, and then he did a breeding with Nick, so I think his was to one of the European line animals, and he sent me one of those, and I put them into a rack and just fed them and raised them up and didn't think much of it. They're both in ARS 70 tubs.
Okay.
Feed phenomenal. They both eat kind of medium rats, and I'm of the opinion that age is much more important than size. And also, whenever I don't have a whole bunch of people to compare Dunn's python size to, I don't really know, are mine bigger than yours or smaller than yours and so on. So I cycled mine just the way in the same rack that had Antaresia in. So that I had some temperature cycling, not a lot, but I did a lot of food cycling. And I put the pair together and didn't see any courting, didn't see any breeding at all. And then I opened the tub one day and I saw them locked. And then 2 weeks later, the female ovulated. So if you look on my Instagram page, I think I got a picture of it. It would have probably been back in January or February. But I pulled up in the tub and the female looked like she'd swallowed a football. She was enormous. And she did the whole process as you expect. She did the belly inversion, dropped 5 eggs. And it's funny because whenever I took her out, the same day, I think one of my Antaresia laid eggs as well. And whenever I took her out, it looked like she was sitting on about 15 eggs. I thought they were going to be tiny, but the eggs were huge for the Dunn's pythons. I was surprised how big they were. And she had 5 eggs, and I just incubated them in the same incubator with the Antaresia. And they hatched out in, I don't know, 59 days, something like that. I was away. Where was I going? I was going somewhere. I can't remember where I was going to, but I was away for a week or two, maybe a week. But the day before I left, I started seeing the lines appearing on the egg, from the egg tooth, but they hadn't pierced through it yet. And then, just as I was leaving to the airport, I saw like a little head sticking out of one. I thought that was pretty cool. And of course, what everybody would then do would be pull out scissors and cut the rest of them. But I'm not a scissor person. But then I came back, and then just all 5 were out, and they looked fantastic.
Bright orange babies.
They were 3 orange and 2 gray. 2 silver.
Okay, that's interesting.
So the adults, the male was silver whenever I got him, and the female was red or red orange.
So they're kind of like a large savu then, I guess.
Yeah, absolutely.
Almost.
Yeah, absolutely. Yeah.
Okay.
And feeding them, they were a bit of a pain to get going. And I'm trying to think what I tried them on button quail. I tried them on live mice. I tried them on defrost, and they were just a pain in the ass. And they were not aggressive. So you couldn't elicit a strike from them. And eventually you just had to beat the hell out of them. And I got a really weird way of getting boas and pythons to feed. And that is I find that if I superheat the rodent, but if I breathe heavily on them, you're probably going to make jokes about bad breath, but if I breathe heavily on them, it elicits much more of a strike response. And I think it's like a predator kind of going at them and breathing. They feel that heat and the breath on them, and they very often then get into like a strike pose, and they'll strike.
You breathe on the animals or on the rodent?
On the animal, yeah. Just go up there like,
really?
Breathe on it, yeah. And it works.
Never heard.
Yeah, and it works.
That's a weird one.
Works with emeralds, Amazons. It's worked with Dunn's pythons, ball pythons, Antaresia. Doesn't work with any colubrids, but anything with a heat pit. Pretty well with. Doesn't work with womas because they're when they got a tiny little heat bit. But yeah, it's amazing how, and it's embarrassing. But I've got my Darwin carpets and my womas at a friend's house, and we produced Darwins 3 years ago, I think it was, and they all hatched out, and he was having trouble feeding them, and I was down there visiting, and I said, "Try this," and we went through them, and they all fed just by my maybe it's my breath or whatever, but because of the I think it's that burst of heat. They see it as a predator coming at them, a mammalian predator coming at them, and they just get instantly that kind of shock.
But wouldn't that elicit a strike and release response
It
instead of a
does.
strike and coil?
It does a bunch of times, but if you just keep at it, then they get pissed off, and then they coil.
And don't get me wrong, I've heard all the craziest, stupidest things that make no sense and sometimes work, right? So, but this, I'll be honest, I haven't heard. I haven't heard new ways, but that's new. That's interesting.
Yeah, I don't know how I came up with it, but I've used it for 30 years, and I used it last night on some baby boas that have only been live feeders, and each one of them turned around and took finally last night the last 5 holdbacks.
Really?
Yeah, it's.
I'm gonna store this in the back of my head for if I need to use this.
Don't ask me how I find out, but
see now the more important thing is it more effective like in the morning before you brush your teeth, or is it more effective?
That's a good question, because I find I feed all my snakes either really late at night or really early in the morning, and I find most of them feed best at about between 5 and 6 in the morning, especially my corrales, because they're sitting, they're perching, and so on. But I find most of mine, I've got boas that you can offer at night, during the day or late at night, and they won't touch. But at 5 in the morning or 6 in the morning, then they'll totally take the food. Yeah, it's a trust me. It's I don't know how I find that out, and
I believe it.
But you know what? It's worked for 30 years, and it's worked for hundreds and hundreds and hundreds of babies. So I'm going to keep doing it. Yeah.
That's pretty. Honestly, I might try that eventually.
Right. If you got a problem feeder, just get right up into it.
Yeah, I've got 2 problem feeders left, so I'll definitely give it a shot. I mean, I got nothing to lose.
Cheyenne needs to try that with those inlands.
The depth of your lungs, the back of your throat. It's not blowing lightly on them. It's that hot air expelling onto them. I think is the key.
Interesting. For this food cycling with the duns, what do you mean by food cycling? How did you go about doing that?
I do the same thing for all of my boas and pythons. I feed them from March through to November, beginning of March to the end of November, and then I don't feed them from the end of November through the beginning of March. And but in the final month, the 6 weeks leading up to the end of November, instead of feeding them every 2 to 3 weeks, I'll feed them every week on a small meal. So they get a whole bunch of food going through and cycling through very rapidly. And then I separate them, keep them separate for about 2 months, and then introduce the pairs together. So they just had that time, that kind of, like in the wild, that glut of food that comes in, because it's all cyclical for many cases. I don't do that with my Antaresia. And we don't do it with womas just because they don't really need it. But I find with a lot of other species it works really well. With all the tree boas, it works really well. You can get them to ovulate pretty much any time of the year by putting them through that cycling phase. They don't need temperature differentiation. And I think with the duns, they don't need the temperature cycling. I think it's just the food cycling works for them as well, just like many of the boas. But it's just, I normally feed them medium rats instead every 2 to 3 weeks, male 3, female 2. Instead of that, I just put the female on a weekly meal of a small rat. And
yeah, that's what.
And she rebounded. It meant that whenever she laid eggs, it didn't look like she'd laid eggs. They're not fat snakes at all, and they're ready to go again this year. I'm looking forward to trying them again because they were a lot of fun.
Yeah. See, I cycle my Antaresia both, and I do the same thing, and I actually cycle them down with temperatures. I just think that they're just so willing to breed because they're a small species that is probably predated on all the time, that for them it doesn't matter. They're so, I don't know, they're so like ingrained. It's ingrained in them, right? They want to breed. So
yeah, I think Antaresia really benefit from the temperature cycling, more so than many things. But also, you look at the size of Antaresia. They're everything's a predator to them. So they're as a species they've evolved to live fast and die young, and produce a lot of offspring, in the hope that some survive. So I don't think they need as many of the triggers, but I think temperature does work really well with Antaresia.
Yeah, I've had good success. So going back to the duns, you said that so they hatched out. Some were orange, some were gray.
Yeah.
How do they look as adults from starting from gray and orange? Do they all end up the same color?
Yeah, pretty much so.
Or
Yeah,
yeah,
you'll see some scales. Some scales in the orange ones will stay slightly orange. You see little speckles here and there, but they pretty much look. They're not like as uniform as a Maclots. They're more like you look at a Savu python where it's black and it's got those white flecks along the ventral kind of close to it on the laterals. You'll get more flecking. But between the grey and the reds, I haven't noticed
no. So
any real differences at all.
you couldn't look at an adult and say, "Oh, this definitely came from an orange
Yeah,
neo."
I don't think so.
No.
I'll have to go down and look at them in more detail. Again, they're very food oriented, so maybe the next time I give them a meal, I'll then spend some time looking over them and seeing if I can pick out more of the juvenile colors. But
And
they once they hit about three, the juvenile colors were gone. I was hoping that the female would have kept more of the reds into adulthood, but she didn't. But every python I have is really food oriented. Whether it's the Timor pythons or the womas or the Antaresia or the duns. So I need to sit down and actually look at them next time I feed them. But they're a really cool snake. I like them a lot. I didn't hold any babies back, and I'd like to try and get one or two that were unrelated to this group, or as far as I can get. Some wild caught ones came in last year, the year before that. Somebody offered me a male recently, but it was a little bit. I felt it was a bit too expensive for what it was. But I would definitely pick up another pair if I could get ones that were more that were slightly outbred compared to mine, because I think they're cool snakes. But they're not everyone's cup of tea because they're basically a Maclots but smaller. And
how do you tell the difference between one and a Maclots? Because I know Cody Joe up here got lucky, and he thinks he has one that came in as a Maclot, and then he's like, this is different. But how can you tell the difference? Like, is there any differentiating?
I wouldn't necessarily believe that without genetic sequencing. I wouldn't trust one if it came in as a Maclots not being a Maclots, because there's multiple islands that Maclots appear on. And they can be, there's just like Timor pythons, right? So there's another Timor that might be a different species, but it could also be a Timor that's morphologically a variant just based on the island it came from. So I wouldn't necessarily say that because it came in and it looks different than a Maclots, but it came in as a Maclot, that it's not a Maclots. I don't believe that. I think exporters, Dunn's pythons have a very restricted range. And I think whenever they come in, they're coming in as Dunn's pythons. They're not coming in as Maclots. Whereas Maclots have a larger range. I've never held them up side by side. I haven't actually worked with Maclots in nearly 30 years. And it was only a pet store that I worked in Belfast that had some Maclots. Head structure seems slightly different. Body size is different. Overall body coloration is slightly different in terms of the distribution of the speckling and so on. They just feel like a different animal. But again, as I say, I haven't worked with the different lines, the different localities of Maclots. They're cool snakes. I just don't have the space for them. And again, I like to work with stuff that at least has some kind of market for selling them. And I know that Maclots go up and down in terms of how easy they are to sell and move on. And I don't really want the hassle of having 15 baby Maclots to try to move. Like when it comes down to it, like I'll breed Duns again, I'll pair them again this year, and then I might wait a couple of years to pair them again, because I think their market again is going to be smaller. Right now, the people that are buying them from me are the people that really want to get Duns pythons. But once all those people that really want to get Duns pythons have them, then I think the market's going to be much reduced again. It's going to be much more like a Maclots python in terms of the audience.
Yeah, kind of like a lot of the niche.
Yeah, it'll take a while to get there, though. I think you're a couple of years away.
I think it's going to be a handful of years. There's not enough of us producing them at the moment.
I'd still love to get some. I just don't want to pay the price they're asking for them yet.
Yeah, because I sold them for $2,000 a pair. Which I thought was a reasonable price. And and I could have sold 10 pairs easily.
And see, I think Cody Joe had him advertised at about $1,200 each.
Yeah.
And
That's for the
that's
rings. He had the
Oh,
rings and the white lips at 1,200
okay.
each.
Okay.
Yeah,
Okay.
but that's Canadian, and our market is kind of smaller, but like they're moving,
Yeah.
but it's not. It's a little harder than just like if they were in the U.S., he would have sold out probably the day he posted them.
Yeah. I like ring pythons a lot. I would have them. They're delicate because they're really prone as babies to dehydration.
Yeah,
Just, but then again, so are rainbow boas. Brazilian rainbow boas. If they go for a day without water, as a baby, you come back and there's a little desiccated, crinkled, dried up baby that's dead. So I
yeah,
think ring
rainbows are neat.
pythons. Yeah, yeah, I like rainbows a lot. They're fun to
It
keep and
will.
fun to breed.
We'll have to hit on those. My buddy, I didn't realize it, but he just got more from Don Patton, more red line
Yeah,
Brazilian stuff from Don,
yeah,
and it's like, yeah, I have 28 adult rainbows at my shop.
yeah. It's a bit much. I've got
He's waiting on like 8 females to drop right now.
yeah. That's too much. I like producing a litter or two a year, but I've got
Yeah,
the T negative albinos. I got the T positive albinos, and they're really cool. I like them a lot.
Have you
Um,
seen
the
the pieds that are in Europe?
yeah, I have. Yeah,
yeah.
yeah. I almost
Those
went
are
for a pie this year.
those are pretty incredible. Like you add that
Yeah,
to it. Oh man, I think pied is
yeah,
one of the coolest genes because you still retain the natural colors,
yeah,
and you have like that
Yeah,
weird mutation. And I love pieds.
yeah.
it'll be cool to
I think
see
that'll be my
the
my next
caramel
year's
in it.
project. Yeah, absolutely. I think that'll be my if I add anything next year, that's what it'll be. It'll be a pie Brazilian rainbow boa.
Yeah,
Yeah, I like them a lot. I think the rainbows are just cool in general. I think I'm surprised the Colombian morphs came down in price so quickly because I think the albino Colombian rainbow boas, the T negative, is just awesome. I think the
Yeah,
T positive is
and the
amazing. The
paradigms.
patternless is cool. Yeah, I really enjoy working with those. The characteristics are
I
neat.
I think it's just people don't like Colombians.
I was gonna say, aren't
Yeah,
Like it's
that's
tough. Like
Colombians?
generic.
They've always been
there's a few
cheaper than
people
the.
working with them up here, or well, one guy mainly. But my
Really?
buddy just produced leucistics
Hmm.
and hets, and
Yeah,
they're kind of. It's going to be interesting to see because like some people want like the top dollar, but it's like our market's definitely smaller, so you're probably
yeah,
going to have to come a little down from that unless you're willing to export.
yeah. Leucistics. I saw leucistic Colombians at shows going for $180, $200. There's
Yeah,
again, there's
they're
a
still
limited
trying to get a
market about who
thousand
wants.
for them up here.
Yeah, there's a limited market about who wants a white snake. And
Yeah,
but the albino, the T negative Colombians are just phenomenal, and they hold their color really well. The pattern is really cool.
yep,
But again, yeah, the prices are not. That's why I end up buying a bunch of them because I figured, if I produce a litter of 12 or 15 babies and sell them for $200 each, $300 each, that's still a decent amount of money. And in that
yeah,
price range, that's where things, that's an easy purchase for many people. I find that the snakes that sell well for me are the $200 to $500 range snakes. So if I can produce 2 litters of those a year, like Brazilian rainbow boas, if I could produce,
so they sell all day long
absolutely,
at
yeah,
350 to $500
yeah.
still all day
Yeah,
long.
I do 275 for a male, 300 for a female, and they go
Yeah,
out the door easily. And I figured that whenever the T negative is $500, it's going to be a phenomenal snake for people. Whenever the T positive is
exactly.
$500, and for me, that's worth buying those animals because I think they'll end up being a decent income for those.
I was gonna say, I think it's
Yeah,
important for if you breed animals to find that one species that sells really well, that breeds
Yeah,
easily, and that pretty much funds every single other projects that you have around. And
so
Brazilians
rats aren't
definitely work.
cheap.
Yeah,
yeah,
exactly.
Rats aren't cheap, and buying racks aren't cheap. I just changed out all my freedom breeders and I bought ARS instead because I prefer ARS. They're not cheap, so to be able to do
yeah,
that, Brazilian rainbow boas pay for that. You
yeah.
know, yeah,
It's kind of neat. My buddy, he's making some this year, but John wholesaled him some really nice red line Brazilians,
hmm
and we had them at 5. They're like year and a half old, so you can tell they're. It's like, yeah,
yeah,
this is
color that.
what it's gonna look like when it gets full. And I've seen his
Yeah,
adult pair at the shop, and
yeah,
they are red.
that's awesome.
There's like normal Brazilians, but these things are red.
Yeah,
And yeah, no, like like there's at Toronto, there's people with $150 Brazilians.
hmm
Guy came up and spent the $500 on
absolutely.
a
Yeah,
really nice red line.
yeah. Whenever you
Like
can show
like
that
it
at a show, and
hold your
people see
price
it,
where you kind
yeah,
of
that's
want it.
it. Especially if you're willing to hold them for 6 or 8 months till they get a little bit of size. Brazilian rainbow boas stay small and spindly for about the first 8 months or 9 months, and then they kick in, and then once they add a little bit of girth to them, then you start to see the real colors appearing, and people just love
Oh
them
yeah,
at that point. Yeah, they're great.
the
And
oranges.
they're so easy to breed. You know, they're born, they shed their skin after a week, and then they all eat defrost fluff mice. There's no
Oh
hassle
yeah,
trying to get every single one of them. You know, you don't have to wiggle it. You just set it beside it or on top of it, and you look away and you come back and they're eating their food. It's just amazing. You know,
yeah.
it's so
I hope my
easy.
buddy's het aneries prove out. He has a pair of pos het aneries because there
Yeah.
was a guy out in BC that had an anery that
Hmm.
bred a bunch of stuff 10, 15 years ago.
Yeah.
And then it's kind of sprinkled around, and he ended up with a pair of pos hets that are finally old enough to breed. So
Yeah, that's
that
cool.
litter should be happening soon.
You,
If it proves out, he's like, I'm keeping everything.
you know, Shan had aneries that were Brazilian rainbows. He ended up
Oh
selling
wow,
everything out, but
he
he
should have
hadn't.
held on to those.
Well, he did. He bred them for the past like 20 plus years, and that was his moneymaker
Yeah.
that paid for everything. He just eventually had enough and moved them all out. But he had some aneries that proved out because he had offsprings of them, and I think a couple of them went to his lamb.
That's awesome. So
Yeah, I don't
my
even know
T,
if he has them anymore.
my T
You
negative
sure?
male is possible het aneurystic, but I've got adult T negative or het T negative that are also het aneurystic. So hopefully
Is the
my albino will prove out to be het and will produce some aneurysticks.
that'll be cool.
Yeah.
What is the caramel? Is the caramel the T negative or is the
positive.
caramel the T positive?
T
Because
positive, I believe. Yeah.
because it looks like a T negative to me, just
Yeah,
looking at it. But
there's different people. Like I've seen people list caramel animals. I just go by the lineage. You know, so there's the European T positive, and then there's the outback reptiles T negative, and they're night and day different animals. You know, the T negative is that white and yellow animal, whereas the T positive
Yeah.
is the orange and pinks and stuff like that. There, yeah, and
I
they're
assume Dawn
they
has
seem to be very variable.
yeah.
You know,
Because
so
I think Dawn's had that, I guess, the caramel line for
yeah.
who knows how long, probably over 10 years since whenever it came out, he probably bought some of the first ones. And only recently has he actually shared them to people.
Yeah.
But
They're
I have a
quite
feeling,
improved.
I don't know how those are actually going to sell as hats up here. Because I know, like, they're probably going to try to sell pairs for $2,000 a pair, but when somebody has produced 40 babies,
yeah,
who's going to want 40 het? Like within
again,
a generation, they're going to drop down to $500.
well, that's exactly it. You know, and I think with many of those projects, you have to realize that there's a price point they're going to come down to, but it's not a bad thing, right? So
Yeah,
you spend $2,000 and you buy these, and in 3 years' time, you produce 20 babies from them. You're going to sell those 20 babies for more than $2,000.
I was going to say that's
even if you wholesale,
never an
you're
issue.
still going to make your money back.
No, that's it. You know, I think that the problem is you spend 25,000 on an animal, and then you're hoping to, at that point, that's too much risk for I'm not going to do that nonsense.
Yeah, but usually the math works, right? As long as you spend like a reasonable amount, it's just better when you pick a species that will not depreciate. Like if you buy a diamond python, you know that when you sell it, they're going to be the same price, right?
Yeah.
But if you buy a Brazilian rainbow boa, for example, that is maybe caramel, even if you bought it at $2,000, $3,000, $4,000 and it's worth $500 in one clutch, you make your money back. So I don't know. To me, it's if you calculate it well, usually it's worth it.
Yeah, you're exactly right. That's it. And as long as you have a decent breeding group where you're not reliant on one animal, and I think it all works out like that. There, I'm not trying to become a millionaire. I like to pay for my hobby, and
exactly.
get a few extra things, and I found out that Brazilian rainbow boas and Colombian rainbow boas are really good ways of doing that. Emerald tree boas are great at doing that as well. Emeralds. I sell captive bred emeralds now for more than I ever bought them for. 20 years ago, and I got a list of people that really want them. So.
Yeah, that old Bill Hughes episode of I listed them for $750 and they sold all within like 20 minutes.
The first ones that I produced, I thought
that was funny.
Whenever I first bred them, I thought, I see them on Morph Market for $395. I'm gonna advertise them at $1,500 to people. They've contacted me and they said, yep, and I sold them all, every single one of them.
Yep.
And I found a phase. People are like, how much? And I'm like, $2,500, and they're like, all right, let me know. And You get a good
Yeah.
captive bred animal, it's a totally different story than getting wild caught.
I think there was a shipment the other year of, I think, basins and it was the basins, the northerns, and the caiman lizards. I assume from somewhere in South America in like a captive facility,
Yeah,
and they were selling as if they were captive bred in the U.S.
yeah, I saw
basically.
because some of those came into the U.S. as well. Some of the basins.
Yeah,
And if I remember well, that every single person that got those, every single one of them died, didn't they?
I heard that in the UK or in the
It's
US as well.
yeah, like
Yeah.
the
that makes
people
sense.
spent
I didn't hear about
thousands.
that, but that
Oh, I
makes
did
sense.
from everyone that I know that, and some people spend like 20, 25K on them, and everybody lost all their animals.
Yeah,
So
Yeah, the
all these
basins were
bloodlines in
insane.
and stuff, you know. And yeah, that's
Yeah,
horrendous. Yeah, the basins are just. I don't know why the price is so high on basins,
yeah,
considering they breed almost easier than northerns, and you know
yeah.
it's kind of remarkable.
yeah, but
I
not
think the
many people
caiman
have
lizards are
them.
still doing all right, but I guess those aren't.
Yeah, yeah,
I know a couple of guys with the caiman lizards that are doing pretty good, but
yeah, yeah. They're pretty cool.
yeah, it's like it's like Indo stuff. Technically, it should be captive bred. Legally,
Right.
it should be captive bred, but
Mm.
we all know that even if it is, it's still being kept kind of semi outside, and it's kind of iffy in terms of just husbandry and that. So
Yeah, yeah,
even if it is, you have to hope that they know what they're doing.
yeah. Right. That's it.
Alrighty, Dominic. I think maybe we should close it out here.
Cool. You're
But
muted.
you're on mute.
You're still muted.
You got to do
There
it
we go.
once every show, right? I was gonna say I'm
Oh,
I'm
yeah.
good with it. I got to get up in 5 hours to go to work, so it's not a bad thing.
Yeah. Yeah. All right.
All right, well, Warren, really thank you. I really appreciate it. And if ever you're willing to come back in a few months, maybe 6 months, a year, we'd love to have you back. That was an awesome show. Like honestly, like full of information. I loved it.
Yeah, anytime. Just give me a shout. I'm always happy to.
Awesome. Thank you, Warren.
Yeah, no
If you
problem,
just
man.
want to stay on for a minute just to make sure it fully uploads.
All right, that sounds good.
And is there any? Did you want to plug any of your information if somebody wants to get a hold of you?
I think the easiest way to find me is just on my Instagram. Just look up Boa, and it's a terrible name, but because I do so many pythons and all this stuff. But you'll find me on there, and if you need me, just message me through it.
All right. For myself, Dominic Cablano, Dom's Reptiles. For Michael Gillian on Facebook. And go ahead and close it out, Michael.